Hi everyone, very good afternoon guys. I hope I'm audible and visible to all of you, and welcome to the first part of Microbiology LRR. I hope all of you have gone through and revised the pathology 2 parts, and now we have similar 2 parts for micro. I think we know the drill by now. That first I'm going to remind you once again about the INI-CET Champions exam that is coming up from April 23rd to 27th, and hoping that all of you will not be scared of this like you are scared of other grand tests, and you will take it with the correct spirit, and you will go forward and attempt it. Also, we have added audio explanations this time to all the questions, so that you get that personal connect with the educator, and you're able to solve your doubts, because I think all of you are just solving grand tests, but the doubts remain unsolved forever. So I think the audio explanations will take care of that, and it's exactly similar to the exam pattern of 4 sections. Also, coming back to the drill, which I always tell you, that LRR alone will not suffice for any exam. It is a trio that you have to go ahead with. You have to first go through rapid revision, then you have to revise that in a concise manner from LRR, and then you have to go ahead with the Q bank. So don't miss out on any of these. It's a trio which is going to help you solve this exam. Obviously, this is a common one for all, but like I did for pathology, I gave you extra topics for INI-CET, which I will do tomorrow as well at the end of microbiology. And you know about the PDF. We have also started uploading the PDF on the app. So you will get the PDF on the app as well. So I think that should not be a worry. This means you will not ask me again and again for PDF, because your focus should be on studies and not on just downloading a PDF and never getting back to it. Now, microbiology is particularly a subject which everyone thinks we will listen to it later on at a 2x speed and we will master it, but somehow we procrastinate and we never do that. So irrespective of whether you find it boring or you feel like quitting, this is a subject where you're not allowed to quit. I don't give you that option that baad mein dekh lena, that sort of a situation. I don't want that should arise. I want you to finish it off today, one entire set of reading this LRR PDF with me right now. And then of course, one set of revision you will have to do later on, right? Okay, so part 2 is also tomorrow. I think everyone knows timing is the same. Today it's at 12, tomorrow it's at 12. So these are the 2 parts that we have. What is the plan for today? I think let's come down to the real thing. We always have to start with general microbiology, which I will. The first most run-of-the-mill question will be gram stain, Zn stain. Wahi har bar. Starting to wahi rehta hai. I've added a few culture media questions also this time. Thereafter, we'll be going to mycology. Thereafter, we'll be going to parasitology. Now, that's too much to cover in one session. So I will try my best to cover it. Just in case we miss out on little bit of helminthology, we can always take that up tomorrow. But at least let us try that. It's going to be a 2.5 hour to 3 hour session. We will sit at a stretch, because we have to sit in the exam also for this much of time, and we will do MCQs plus tables. This is going to be a combined kind of a session. So I think that is okay for everyone. We are all set to go. Someone was saying Hindi. I'm very sorry, although we have English explanations and everything in English on the app, but this is a common session for every student, and I will have to conduct it primarily in English, because that is the language which everyone understands. So here I cannot go in for English and English separately. I will have to go in for English so that everyone can understand, and students who understand English, they can also understand English. I think that should not be a problem. Let's get going. We always have some questions which I begin with, but I've changed the order this time. The first question I've taken as a screenshot from the version X QBank, and that is that what is MIC, that is minimum inhibitory concentration of an antibiotic. You have 4 options given in front of you. I hope with the word minimum, you are only thinking of the lowest things, minimum, and you're not thinking of the highest concentration. As always in the exam, ma'am, 2 options are easiest to rule out. Common sense, non-medico can also rule out. Two options is where the real game starts, and most of you have told me that this is B, that is the lowest concentration that will inhibit the bacterial growth. Exactly, because the name says minimum inhibitory concentration. So this is not how easy the question is going to come. I am going to now tell you that how are you going to test for this minimum inhibitory concentration, and that is done by this method, which I know most of you don't study because main videos stuck, you don't have the strength to go in microbiology. But this is how you're going to study, and this is known as the dilution method, which is said to be one of the gold standard methods for knowing MIC. So please note, it is known for MIC, and we don't have to know the procedure, but I just need to know that I am going to grow an inoculum of bacteria. So let's assume you are dealing with the most common patient of urinary tract infection. Okay, some of you said that the volume is an issue. I don't think I've tested it prior, and I believe everyone voice is not good. Somehow there is no change in the settings from my end. Everything is exactly how it was. I don't know if it's still coming across as a problem. Some of you have said that, okay, if the voice was a problem, they rejoined within 5 seconds and it worked for them. So I think if it's that's the thing, probably a connection or a technical issue with YouTube. But I think if you just quickly rejoin, it should be good to go. Coming back, I have taken, let's say, a urinary tract infection. I've got a urine sample from the patient. I want to know which antibiotic to give and at what concentration to give. So basically, I will be growing it with different dilutions. This mentioned over here are the different concentrations of the antibiotic that I have added. Now you need to understand that at what growth will the bacteria stop growing. So I realized that the bacteria are growing at a 0.5 microgram concentration, they are growing at a 1 microgram concentration, they are growing at a 2 microgram concentration, but they have stopped growing at a 4 microgram concentration, which tells me that minimum inhibitory concentration is going to be 4 microgram per ml, and that will be the MIC for this antibiotic that we used. So now coming back, this is the minimum inhibitory concentration. But are there other methods? Because now I'm going to make you answer another question, and thereafter I will start my discussion. What statement is not true regarding Kirby-Boyer method? Number 1, do you apply 4 to 6 antibiotic discs for every plate? Factors such as nature and composition of the matter or the medium also influence. Incubation is usually done overnight. Disc concentration does not affect the zone of inhibition diameter. Okay, so this is right. So most of you have come out with C and D as the options. They have said which of them is not true, and I will go with the majority, and let me explain this to you. I am talking about Kirby-Boyer, and I've started today's class with this because this is actually what is, I would say, the most trending hot topic for every exam pattern, because we are in the era of antibiotic resistance, as all of you know. So when I'm talking about antibiotic resistance, or I want to find out in a case of, again, urinary tract infection, the urine or the patient will, what antibiotics should be advised to this person. He would respond to which antibiotics. I do the Kirby-Boyer disc diffusion method. Kirby-Boyer are names of people, so that's okay. Disc diffusion will tell me that I will be using discs of what? Discs of antibiotic number 1, antibiotic number 2, antibiotic number 3, so on and so forth. So before these discs, I have added some agar, like you always add blood agar, chocolate agar. Over here, you have added CAMHA. They ask you for Kirby-Boyer disc diffusion, what agar is used? What is CAMHA? It is cation adjusted Mueller-Hinton agar. Sometimes they write CAMHA, sometimes they only write Mueller-Hinton agar. Whatever is written, it's the same. They have used Mueller-Hinton agar. After that, they have put whatever urine sample was there, they have put that entire urine sample on top of it. They have covered it. How have they covered it? Like a lawn. That's question number 2, that how is the sample put? It is going to be like a lawn culture, like a garden, like a lawn. Everywhere urine has been spread out. Now they have put antibiotic discs. Over here, antibiotic discs that have been put, they are put at least at a distance of 25 mm, so that there is enough space for everyone to work. Finally, there is an incubation that is done overnight, approximately 37 degrees. And now you're wanting to see whether the antibiotic could work or not. Do you see bacteria has grown everywhere, but the bacteria has not grown around the antibiotic? This means this antibiotic is sensitive. Can I say the bacteria was inhibited over here? This is known as zone of inhibition. This is known as zone of inhibition. So please note, zone of inhibition, if it is seen, this means the antibiotic has has worked, antibiotic is sensitive. So over here again, I can see bacteria has not grown around it, antibiotic sensitive. Bacteria has not grown around it, antibiotic sensitive. On the other hand, if I come to this particular antibiotic, bacteria is growing exactly on its face. This means this antibiotic is not working, and that means the antibiotic is resistant. So simple, if you see a zone of inhibition, this means the antibiotic is sensitive. But did it tell you the MIC? No, ma'am. Over here, it could not tell me what concentration of antibiotic is used. So did it tell you the MIC? No, but at least you can understand that if I would have used a greater concentration of antibiotic, the bacteria would have been inhibited more. If I would have used an even bigger concentration, the bacteria would have been inhibited more, which further tells me that yes, the concentration is going to affect the zone of inhibition diameter. If you use a very strong and a high dose antibiotic, the inhibition will be more. So it's not that the disc concentration does not affect, it affects the zone of inhibition that is formed around it. But one quick question, one disadvantage, ma'am, it did not tell you the MIC, but what will tell you the MIC? Perfect. Everyone started telling me that epsilometer test, which is also referred to the E test, is going to tell you the MIC. How does it tell you the MIC? Because firstly in the exam, what do you use? Ma'am, I use the same CAMHA, cation adjusted Mueller-Hinton agar. I use the same lawn culture, urine is spread everywhere. But instead of using antibiotic discs, I have used E test, a strip. Can you see E is written on top of it? So that is the epsilometer test. But if you look carefully, whatever, let's say there is an antibiotic on top of it, but on this I have got concentrations of the antibiotic. So now you will see that again there is a zone of inhibition that I can see. So antibiotic is so sensitive, but at what concentration did the zone of inhibition start? That will also tell me the MIC. That will also tell me the MIC. So I think that is okay with everyone. If you want to test for MIC, do epsilometer test. Otherwise, what is very commonly done in the labs is Kirby-Boyer disc diffusion test, but that does not tell you the MIC. But you know what is still, see, some things are old school, is always old is gold. Still what is considered as, you know, what I would say as the gold standard is the dilution method for knowing MIC. But again, it's very cumbersome with so many test tubes, so not many people do it. But again, that's the OG is what I would say in this entire situation. Someone asked me, like Dr. Himanshu asking me about Stokes method. Stokes method and Kirby-Boyer disc diffusion are the same. This is only important for INI-CET students. Stokes method and Kirby-Boyer is the same. The only difference is in the Stokes method, I have added one control strain. You know, you're doing the test, but do you know if you're doing a right test or not? Have you performed it properly? Is this test even valid? To know the validity of the test, I have added one bacteria which I know is sensitive, which I know it will get inhibited or killed by the antibiotic. So I have added a control strain, and that's about it. Otherwise, there is not much of a difference between Kirby-Boyer and Stokes method. Finally, we are done with all of this, and now we go on to the next question. We have some people who are spamming the comments, and I just feel that it's a very sad thing to do, so don't do that. If you still want to continue to do that, enjoy yourself. We don't have so much of time to pay attention. Now coming to the second question. Now what is this? So which bacteria we add? Dr. Fatima, those are control strains depending on which sample you are dealing with. Okay. Now coming back, let's start with this. Which staining sequence should be used to stain the smear of a sputum sample? In India, sputum sample TB has come in everyone's mind. ZN stain has come in everyone's mind. That works at a construction site with fever hit hemoptysis. Ma'am, obviously we are dealing with ZN stain and we know the correct answer is D. There are two stains, ZN stain and gram stain that we need to do. So ZN stain के लिए एक mnemonic, gram stain के लिए एक mnemonic. Revise the entire topic with me right now. I'm giving you 5 minutes. Today I've got an even tighter time boxing frame that I've done for you, because micro matlab if I sit with it I can get bored to hell, and I don't want that, right? So I want everything to wrap up very quickly, but I should finish everything also. So I have to be a little more a little more fast, and I have to be a little more attentive today, because otherwise we will all sleep off with this subject. Starting with ZN stain, yes, class has asked mnemonic, excellent. So now tell me the full form. The first thing that we are going to add is going to be carbonyl fuchsin. What color does carbonyl fuchsin give? It gives a red color. That is why you will notice that the organism has come out to be red in color. All the TB is going to be red in color. Thereafter we have class has, H for heat. Heat is going to act as a mordant. These are all MCQs that I'm telling you. Mordant matlab fixer. It will make sure that the red color gets fixed over there. After that, we are going to have A for acid alcohol. Which acid is used? Sulfuric acid. Very good. And that's the decolorizer, because this will try to take the red color out. But TB is TB. TB is acid fast. TB will not leave the red color. Now TB has come red. So what will you do to the background? To the background, I will put methylene blue. So obviously it is going to give a blue color. So that is why I asked, class has asked mnemonic. This is the best statement going over here. So the background comes blue. Now what they'll do in the exam, they will not give you this photo and they will give you a fill in the blank saying organism is dash and background is dash. So organism is red and background is blue in color. I hope that's okay even without a photo. But the story is that what are the concentrations of H2SO4? Everyone has started telling me that ma'am TB socha tha. So for TB it is T20, 20, 25%. These days the latest textbooks have started saying 25%. Then for M lepra we always used to study 5 alphabets. So 5% H2SO4 is what is going to be used. Also when 5% H2SO4 is used, that is known as the fight for acostain, another previous year question. Now coming to nocardia and legionella, over there 1% of them is going to be used, 1% H2SO4. Latest question, what is modified Zn stain? This is not the cold Zn stain, guys. Modified Zn stain is done for nocardia when I'm using how much of H2SO4? It is 1% H2SO4. Please notice this, and this could be a question. आजकल का ना थोड़ा सा new trending वाला category का question है. Coming to the spores and the head of the sperm, the ones that look like S, whatever numbers you can think with S, I can think of 5, I can think a 2, so anything around it, very minimal concentrations, 0.25 to 0.5% is the concentration for spores and head of sperm, which means so many things are acid fast. Yes, my nose is cold and hot. Why did I learn this mnemonic? Because for Zn stain, if I remove the heat, what would that be known as? Then if the heating step is removed, that is known as the cold Zn stain. Is there other name for cold Zn stain? It is also known as the Kinyon stain. Any other name for the cold Zn stain? It is also known as the Gabbett stain. So cold Zn stain is known as Kinyon stain, in which you have just no cold, you've not frozen it, you've just not done the heating procedure. Now for INI-CET students, even NEET and FMG, it's important. Heating nahi kara, so you've not done the mordant. Next question, how will you now replace it? What are you doing instead of the mordant? If there is no mordant, then you will have to make sure that you've added a lot of red color. Red color is what? Carbol fustian. Carbol fustian is a phenol. So basically, when you're doing this kind of a situation, you will increase the concentration of phenol because you know that you don't have the mordant with you. Now, because you are dealing with so much of heat and cold, the mnemonic is also heat and cold. Fill in the blanks today to save time. I have written everything taki we are not wasting time in writing. I have done the writing part in main videos RR. Today you will revise with me. When I say my nose is cold and hot, what is my? My is going to be mycobacterium. Any mycobacterium, TB, leprosy, atypical, kuch bhi chalega. Nose, no for nocardia and legionella. I hope you remember nocardia, 1% H2SO4. That is modified Zn stain. My nose is reminds me of the is, that is isospora, and then along with that its cousin brothers cyclospora and cryptosporidium. Why are these three cousin brothers? Because together what are they known as? Ma'am, these are the coccidian parasites. Maybe you will teach us today. And for coccidian parasites, which kind of Zn stain is used? For cold Zn stain is used. What is the other name? It is the Kinyon stain. So that makes us Shah Rukh Khan. We do all Kiran kind of things. So we have coccidian parasites are also known as cold Zn stain, are also known as the Kinyon stain. So ye sari cheeze come together. Now if you've written my nose is, you are not forgetting the spores and the head of the sperm. That is also going hand in hand. So sperm head will come and spore will also come. My nose is cold and hot. HO is hooklets of hydrated. We are going to study this today as well. And T for, I think this kind of a question will come. Which Taenia egg can be Zn stain positive? We have two Taenias. Taenia solium. Solium is the one that goes into the brain, neurocysticercosis, not that one. Taenia saginata. Saginata egg can be positive for Zn stain. So my nose is cold and hot. Every alphabet has to be learnt. Don't filter it out. If you've done the Zn stain without a question, I've done this today in my micro I've got one question, but I can't keep you solving questions, then we'll be on one topic for 1 hour. So with one question I have covered all the PYQs and the important points of that topic in that one question. Now if I ask you, instead of the Zn stain, the gram stain, jaldi se in 5 seconds tell me, it is known as the come in and stain. This I think everyone remembers. What is come in and stain? Number one is going to be crystal violet. Tell me what is happening at every step. What is happening at every step? So crystal violet, ma'am, violet, purple, everything is purple. Gram positive is also purple, gram negative is also purple. Then I add iodine. Iodine acts as what? That is the mordant. Ye yahan ka question. Mordant means fixer. Now mordant is going to make the purple color even more. Now also gram positive is purple, gram negative. Starting mein everything is purple till the time I have added the alcohol or acetone, because that is the decolorizer. Now the color will go. But gram positive, see, I'll tell you the cell wall, it has such a thick cell wall, it doesn't give the purple color. It says I will keep the purple color. Gram negative organisms get decolorized. So if they ask you that after adding crystal violet and iodine, what are the colors? Everything is purple. But after adding decolorizer, now only gram positive are purple and gram negative is going to be colorless. And finally coming to come in and stain. S for saffron in red, because if you had made gram negative colorless, you will have to add a color onto them. So finally at the end, if I ask you what is the situation like, gram positive has come out to be purple and gram negative has come out to be red. So that is going to be everything important. Now many of you are asking me, ma'am, tell negative stain this and that. So abhi what I'll suggest, keep all your doubts of miscellaneous things thodi there, 2-3 hours you will have to keep patience, because maybe this is not the right place to study negative stain. But when I go to cryptococcus, of course I cannot complete it without teaching you negative stain. So abhi ke liye just don't go into that FOMO ke ye reh jayega wo reh jayega. It is my responsibility, I will make you study it. 10 minutes extra lagega, pad lenge. I am in no hurry, I have no work, I've taken off. So I can sit for 5 hours also. My concept is finish the syllabus, but at the right time we will study the right thing. And Dr. Fatima, we will come to percentages, but वैसे it is a confusion, but I will come to that. Just give me abhi na, just go with the flow, be with me. I know where to ask you which question and how to integrate it. उसके बाद अगर बचेगा कोई topic, you ask me after 2.5 hours, ठीक है? Now coming back, what if everything is gram positive and gram negative? तो कुछ तो होगा ना मैम cell wall में which are neither gram positive nor gram negative, that is gram poor. Gram poor मतलब कौन कौन से? They are MRCS. They have some or the other problem in their cell wall that they cannot be gram positive nor gram negative. So my for mycoplasma, which you guys keep telling me, ma'am, wo fried egg wala, that is gram poor. Rickettsia, chlamydia, spirochetes. Tomorrow we have bacteriology. Tomorrow I will teach you these 4 which you always leave because these are the last 4 chapters of bacteriology. These 4 are mycoplasma, rickettsia, chlamydia, spirochetes. These are gram poor, and tomorrow I will ask you why they are gram poor, because bacterio day is tomorrow. All the stains are done जो अभी के लिए important है. Now I am going ahead and asking you questions that they will give you from labeling of microscope. So 3 microscopes are asked in the exam. This is the usual light microscope that all of you know. Image number 1 labeled will be what is this? What is this gold sa thing? Because I label some things for you. Obviously what you have at the bottom is the light. When the light will come, the light will go through. Excellent, ma'am. This is known as the condenser. Amazing, guys. Condenser does what? It condenses the light. When it condenses the light, where will it condense the light? On this stage. What is the stage? Stage is where the hero is, ma'am. The hero in pathology and microbiology is the slide. So you have kept that TB wala slide, cancer wala slide on this stage. They ask you, condenser is below the stage, above the stage, on the stage. You can see it is going to be below the stage. After that, what we notice is that there are some objective and eyepiece lenses. Matlab there are some lens system, because it's a microscope. It has to zoom in. Yeah, there is some voice problem which many of you are telling me. Let's just, I think this is not at my end, but some technical issue with YouTube. Let's continue for 5-7 minutes. Tab bhi rahega, then we'll think of what to do. Abhi there is actually nothing that we can do because it's a YouTube issue, I believe. Coming back, when I talk about these two lenses, objective and eyepiece lens. Object wala is close to the object. Eyepiece wala is where I am sitting getting close to my eyes. So eyepiece, many of you told me, ma'am, it has a 10 times zoom in magnification. That we know. When I look at objective lens, it has 4 lenses. Tell me, excellent. Number 1 is going to be 4x. 4x means what? That is going to be scanner. Then low power will be a little more, that is 10x. High power will be a little more, that is 40 times. And if you put oil and view the slide, that is 100x. So rather than learning it like this, I think it can be 4, 10, 40, 100, right? 4, 10, 40 and 100. That is a better way of learning this. So these are the 4 objective lenses. How do you, don't tell me that question, ma'am, hame maximum magnification ratta hua hai, that is going to be 1000x. I'm not asking you that. How do you calculate total magnification? It is going to be a multiplication of these two. Eyepiece multiplied by objective is the total. So if they say that you are currently viewing the slide on high power, this means you are viewing the slide on 40x. Multiplied by eyepiece will always be 10x. What is the total magnification? It is going to be 400x. But if I ask you what is the maximum magnification, that question everyone has ratta lagaud. Maximum magnification is 1000x because maximum is 100. 100 multiplied by 10 is obviously going to be 1000. So this is, I think, a very run of the mill ghisa pita question which you know. This is basic light microscope. Solve all 3 PYQs which I've taught you in previous LRRs. So now the story is that I have incorporated all previous INI-CET, NEET, LRRs in one. You don't have to run here and there to view 400 LRRs. Everything is over here only. So if I ask you, the microbiologist wants to study cells, the senior advised him to use a light microscope. They want the sequence. Please read sequence from where to where. From eyes to the light source. From eyes to the light source is what they are asking you. So obviously the first thing near the eyes will be eyepiece. After that comes the objective lens and then comes the condenser. So yahan par na the story is that this is also a correct sequence, but they have asked you from where to where is what you had to study. Asking you another question from the previous LRR, where is the condenser present? Above the stage, at the stage, below the stage? That is going to be below the stage. Abhi padha ma'am. You know that. One more question from the previous LRR, that is, in the light microscope, what function does the condenser serve? What is the function of the condenser? You said it is present below the stage. So what will it do? It will focus the light on the stage where the sample is kept. Below the stage, so focus the light on the stage, on the sample wherever the slide is kept. All 3 PYQs of this topic are done. So this means today LRR में PYQ तो मैम हम कर ही रहे हैं. Some potential questions you are telling us, updates you are telling us. हमारे लिए क्या बचेगा बाद में? You will have to solve the Q bank on your own also ना. The other miscellaneous questions is your homework. Coming to the next, light microscope done. अब now let us finish all the PYQs of fluorescent microscope. This is a fluorescent microscope. How did you identify something is shining? This is fluorescent. Number 1 question, which light is used? Start answering all these questions. It is going to be the ultraviolet light. After the ultraviolet light goes, what is this special mirror which no other microscope has? It is going to be the dichroic mirror, which is amazing. After this, ma'am, whatever sample you have kept, TB ka sample, malaria ka sample, on top of that you are going to put a dye. So you will tell me the names of all the fluorescent dyes that we have. For TB, TB ke liye kya hota hai? Your day, guys. It's not my day today. I have done a very simple thing. I have made a PPT, I have written all the answers, and then I have hidden all the answers because today is your test. You will, like, you used to sit with your mothers or your parents when you were in school as school kids, and they used to take your copy and make you answer things and ask you. That is exactly the role I am playing today. With micro, there is no other way of learning. Perfect. Auramine, rhodamine. Are you sure you want to call it auramine and rhodamine? You don't want to call it rhodanine because rhodanine I taught you day before yesterday. That is the stain for copper. It is different. For malaria, kya hai? Malaria ka fluorescent stain is acridine orange. Very good. For fungus, it is calcoflower white. Very simple. Floor. The word only tells you. It's the fluorescent stain for fungus, calcoflower white. Now tell me, INI-CET students, flow cytometry also uses a fluorescent dye. What is the name of that fluorescent dye? It is FITC. Yes, that's the name of the fluorescent dye for flow cytometry. For flow cytometry, it is going to be FITC. So good. So UV light aega, dichroic mirror is going to come, dyes will come. Now if you've done so much, tell me the principle also. This is a previous year question again. In fluorescent microscope, the UV light that is going is actually of a shorter wavelength. Number 1, but my eyes will not be able to see it because it's UV light. So I will have to actually convert it into a longer wavelength for us to be able to see it, and that's the principle. Smaller or shorter wavelength converted to longer wavelength. सारे PYQs हो गए. Fluorescent microscope done. Identify this microscope, which you know, ma'am, the microscope that looks like a spaceship. हर जगह से closed. Which is that? It's looking like a spaceship ready to launch. That is an electron microscope. Tell me the scientist who identified ये electron और light दोनों. Ma'am, E for E, that was Ernst Ruska, the two most important scientists for microscope. And light microscope, I think everyone knows, is going to be Leeuwenhoek. So E for E and L for L is how we had learnt it. Now coming back, because ma'am you said this is all spaceship, why have they made it like this? Because over here you're using electrons, and you know electrons can only travel in vacuum. If you make electrons travel like this in air, they will be lost and they will not be able to show you the result. So for electrons you need vacuum, and hence it is all closed. Whereas for a light microscope, because you were using light can travel in air. So for light microscope you were using light and air. ये तो common sense question है. Question आएगा, what is the fixative? This I taught you in patho also. Last time I'm teaching you in micro. For light microscopy the fixative was perfect. 10% neutral buffered formalin NBF. And for electron microscopy the fixative is 2 to 2.5% glutaraldehyde. So can I say एक का formaldehyde formalin, एक का glutaraldehyde? I think this is also settled. But now they will ask you later on that what composition, this is for INI-CET students, what slide is being used. I mean, how can we forget second prof trauma, a glass ka slide milta tha, and anyone who broke the slide was always given that the torture that we will fail you in the exam and etc etc. So glass ka slide was only for a light microscope. For electron microscope we usually use metallic slides, and usually they are made up of copper but can be made of others also. Is that okay with everyone? Now coming to the next. अब जब इतने microscope पढ़ लिए तो why not one more? What is this microscope? The one that looks like a complete black background. Of course it has to be a dark background, so it is a dark field microscope. Now they will ask you, what kind of a light have you made in a dark field microscope? We are not using the regular light. We are using the regular light, जैसे bulb वाला होता है, but in that we are also adding certain extra plates, because of which it becomes a reflected light. Now why do I want light to reflect? What kind of structures am I planning to see? Ma'am, whenever something is very very thin and you can't look at it under a light microscope, you use this. What is very very thin? Number one, flagella. When a bacteria has flagella coming out of it for locomotion, for movement, flagella very thin, cannot see, use dark field microscope. Second, any spiral bacteria like spirochete, you can't see it, very very thin, very spiral, very thin, anything that is thin. So they might not write this. They might say that there is a very thin structure of a bacteria which you want to visualize, but you are not able to look at it under a light microscope. Now which is the other microscope that will help you? You will say it is going to be a dark field microscope. One more question that can be a twisted question which you have studied but you have not seen it from that point of view. Question कैसे आ सकता है? What if you have a very thin structure and they say that अरे availability तो is only of light microscope. ये तो you are not that rich, you don't have this dark field microscope in your laboratory, but you want to see the thin structure. So ma'am, if something is very very thin, then you will make it thick, you will make it mota. What will you do? You will start depositing some things around it. उसके आसपास ना you start depositing a lot of silver. When I will deposit a lot of silver around it, that silver is basically going to make that thin structure very very thick, and then I will be able to see it on a light microscope. Very good. That is known as silver impregnation. And now I have made that thin structure thick, and now I can see it on a light microscope. So number one, you will try dark field. If that is not there, then thin ko thick kar do, and you can still see it under light microscope. Have you understood this concept? Can I go forward to the next question, where you will identify this cell wall for me? Is it a gram positive bacteria, a gram negative bacteria, an acid fast bacteria, or a bacteria with atypical cell wall? Whatever bacterial cell wall is written over here. And yes, okay, many of you are saying that is going to be gram positive bacteria. Achha, why did you say itna sa chota sa photo mein, why did you say gram positive? Firstly, ma'am, no one could read this NAM-NAG, but we assumed कि NAM-NAG to hai, and anyway bacteria were mentioned. But when I saw these NAM-NAM molecules, these green dots, whenever I will see this 5-5 dot wala connection happening, that is known as a pentapeptide 5 proteins, pentapeptide bridge, which is only seen in gram positive. So now what I'll do, I'll give you a quick revision of starting from here first. This is the photo that you have to know of gram positive and gram negative cell wall. Same photo aega exam mein, same labelings will come, same questions will come. So when you know that this is exactly what is going to come, then you can't mess up, na? Let me start. When I say gram positive bacteria, you will number one say that, ma'am, it will have a very thick what? Can you see this? Very, very thick peptidoglycan. So very thick peptidoglycan will tell me gram positive. Second, you studied that I can also see that on the top, we will see something known as teichoic acid. Guys, what is it going to be? Teichoic acid? Abhi bhi voice ka problem ho raha hai. I don't see any issue, guys, at my end. This is probably a very unfortunate technical issue at YouTube's end, I believe, because everything is perfectly working at my end. There is no issue. There is no audio concern also at my end. So I really don't know what is going. I mean, is the volume low? Is it cracking? Because everything from the net to the volume at my end is showing perfectly fine, better than usual. Okay, so someone said that they closed YouTube, relaunched it. So yeah, that's what I said. Seems more likely to be an error at the streaming level, which actually we can't do anything about. I mean, no matter how much I may try. So on rejoining, it's better. So I think today it's micro na, no one likes. Maybe YouTube also doesn't, just like us. So then we'll have to just take it this way. Okay, coming back once again, if I say I'll come to the previous slide also, let's just start from here. I'm trying to differentiate between gram positive and gram negative. Gram positive mein kya hoga? There's going to be a very, very thick peptidoglycan cell wall. You can see gram negative mein peptidoglycan is very, very little. Next, in gram positive, you will notice something called teichoic acid. Anyone, what is teichoic acid for? We learn it ticna. Teichoic acid is for ticna, which means it is going to be for attachment. It is going to be for attachment. So whenever the bacteria wants to attach. And second, it is also going to have the pentapeptide bridge. What is pentapeptide bridge? I can't see it over here. For that, I will have to zoom into this and see it. Like all of you were saying, ma'am, humne NAM-NAG to dekh liya tha. But in that NAM-NAG, I want to now zoom in and show you. This is a NAM, this is a NAG. This is a NAM, this is a NAG. NAM-NAG har cell wall mein hoga. Whether it is gram positive, whether it is gram negative. But please note that between the NAM molecules 1, 2, 3, 4, 5, between the NAM molecules 1, 2, 3, 4, 5, if you see these pentapeptide P for P, pentapeptide gram positive, where you don't see these 5 molecules, that is going to be gram negative. So we are settled with this. Positive mein peptidoglycan hoga, pentapeptide hoga, and teichoic acid hoga. I think we are settled on this. Now we come to gram negative. Ma'am, obviously in gram negative peptidoglycan is not going to be there. It's very, very thin. So hota kya hai? What do I see in gram negative? It is lipopolysaccharide. They will ask you, this is a previous year question again. What is previous year question? Labeling is not good. Labeling jitna maine label kara hai, bas utna hi. That is why I have relabeled it for you. Ye kaisa question hua? This is not the labeling that the examiner will give you. Examiner to ye hata ke dega na, wo thodi label karke dega. This is exactly what I have labeled because you need to identify these parts. Coming back to lipopolysaccharide in gram negative organisms. So please note that lipopolysaccharides has 3 regions. Some people learn it as OPA, OCA, same thing. OPA, OCA, same thing, which means region 1 is going to have O for O antigen. Region 2 has polysaccharide. Some people have learned it as polysaccharide core, so they learn it as OCA, but I think core will not tell you much information. I believe OPA will be better. So region 1 is going to be O antigen, region 2 is going to be polysaccharide, and region 3 is going to be lipid A. And this is exactly what they ask you in the exam. What is region 1? What is region 2? So remember OPA or OCA. It is going to be O antigen, then polysaccharide, then lipid A. Now you understand, lipid A and polysaccharide together makes it lipopolysaccharide. So that is also done. Now please note, there is another space. If you look carefully, ma'am, humne cell wall to pad liya, but I can notice that they will mark something like this. Now listen to my labeling very carefully. There is something which is a cell wall that they have marked, okay, and niche they will mark something called a cell membrane, obviously cell wall and cell membrane, but around it, in between them, they will mark something called periplasmic space. Listen to this very carefully. There is cell wall, there is cell membrane, and in between is periplasmic space. What is advantageous over here. Some bacteria keep their flagella hidden. Ma'am, flagella hidden थोड़ी होता है. Flagella तो comes out of the bacteria like this, but that is what I'm telling you. That flagella is hidden inside. That is an endoflagella. Yes, guys, which bacteria? It is going to be spirochetes. It is going to be spirochetes. Perfect, perfect, guys. Amazing. This everyone knows. तो spirochetes में endoflagella कल दोबारा पढ़ेंगे. Coming back all the way, why I gave you this question which everyone was asking, ma'am, once again. अब मुझे बता दो, NAM, NAG, NAM, NAG, NAM, NAG. And in between the NAM and NAM, can you see 5 things? Pentapeptide. If pentapeptide is there, ma'am, gram positive. Simple wala question tha. If this image comes or this image comes, tell me where do you see the pentapeptide cross bridge in the first photo? So which is gram positive? Done. Which is gram negative? The one that doesn't have pentapeptide. Or you get this photo. Every picture on the net, every variation on the net and in the books I have shown you. Now you can't get this question wrong. So if I ask you a simple question which is lengthy but it's the same thing, is that a lab technician is performing a gram stain on a bacterial sample from a wound sample. Under microscopy there are cells having 100 layer thick peptidoglycan layer and purple. Purple to वैसे ही gram positive, and thick peptidoglycan is also going to be gram positive. Additionally, there is no outer membrane, porine, and no outer membrane or porins are seen surrounding the cells. Cells also lack lipopolysaccharide. Gram negative ruled out. This is classical. So see, या तो फोटो आएगा या फिर उसका एक डिस्क्रिप्शन आएगा. Either ways, I think PPPT, if you remember, you can easily solve gram positive. We have given it enough time. We must move forward. We now, of course, have to do a question because you were talking a lot about flagella. We have to talk about the motility once again. So you have to do match the following, which is a very recent question. A question in INI-CET also came. It can come in NEET and FMG. What is the parasite? Neisseria. But ma'am, Neisseria tomorrow we will study is a cocci, and cocci though are not motile. छोटे छोटे round bacteria are not at all motile, but can they be motile? Sometimes yes, and that is because of type 4 pili, which was an AIMS exam question. Belantidium coli for cilia, coli for celi, so this is ciliated organism. Trichomonas vaginalis is of course going to have the flagella, which I will tell you, and Toxoplasma gondii is going to be no particular. I will teach you today in parasitology, no particular organ. So ये हमने क्या क्या बात करी. Usually if they ask you for locomotion, please remember usually it is always the flagella. Flagella, guys, is very thin. You can't see it under light microscope. So which microscope is better? I just taught you dark field microscope is better. So locomotion के लिए flagella, but like I'm saying, type 4 pili are going to be important for two bacteria. Please learn it. One is Neisseria and other is going to be Pseudomonas. For Neisseria and Pseudomonas, type 4 pili is going to help, and for cilia is going to be for B. coli. B. coli is going to be for cilia. Now let me go ahead. कि अच्छा ये तो हमने organs याद करे. What are the different types of motilities that they can ask me in the exam? Each one of them is a question. Hence, I have not got match the following. I have got you fill in the blanks. Starting with tumbling. Tumbling, I always tell you, should remind you of that drunk friend of yours. Tumbling, tully friend, drunk friend, tumbling here and there, and that is going to be listeria. Now another thing that this drunk friend is going to show is going to be differential motility. Differential motility is also shown by listeria, and that says that I will be tumbling, I will be moving and tumbling, falling here and there at 22 to 25 degrees, but I will not be tumbling at 37 degrees. Now what is this? Difference of temperature. So differential motility. How did I teach you that tumbling is like tully drunk? You can afford to be drunk at a young age of 20 to 25 because your liver is going to support you, but you can't afford to get drunk at 37 years of age because your liver is going to give up, your capacity and metabolism will be gone. So please note, tumbling is and differential, dono are answer. Listeria, this is the most recent question that you have. Now coming to darting. Darting means very quick, and that is known as shooting star. So like a shooting star, yes, I only got Vibrio. Guys, are you sure that only Vibrio shows you this? No, I always taught you camping under the stars. Camping, Campylobacter is also going to show you shooting star. Very good. Where do you see corkscrew? Screw to sabne dekha hi hai, nail, screw, right? So that bacteria, it looks like a screw also and it moves like a screw also, and that is known as Treponema pallidum. For those who've forgotten, Treponema pallidum causes syphilis. So ma'am, wo dekhne mein bhi aisa gol like a screw it is, and it moves also like corkscrew. Now coming to twitching. T for T. Twitching is seen in Trichomonas, the one that shows you the strawberry cervix. T for T, Trichomonas. Coming to swarming. Swarming swarming ka photo is in front of you guys. Swarming means aisa concentric round, it's going in circles. Swarming motility is shown by PVCs. Ye to I also know. You will fill in the blanks of PVCs for me. What is P? What you can never forget? Proteus. Now I ask you V. Would you want to write Vibrio cholera or the other Vibrios? Ma'am, go back. Other Vibrios like Vibrio parahemolyticus, Vibrio alginolyticus. Very, very good. Not Vibrio cholera. Vibrio cholera does not show this motility or any motility because I just told you, at least not swarming, because Vibrio cholera shows you what? That fast wala, shooting star motility. So don't mess up over here. Vibrio parahemolyticus and alginolyticus. Coming to next, C for what? It is going to be Clostridium tetani, and if they ask you Clostridium, it is tetani. If they ask you the Bacillus species, that is Bacillus cereus. And lastly, coming to S for Serratia. If I give you the same options and I say there is a patient who's had food poisoning after consuming Chinese fried rice, ye same photo diya, Chinese fried rice consumption and food poisoning, all these options given, that is going to be what? That is Bacillus cereus, which we will study tomorrow. Bacillus cereus causes Chinese fried rice ka food poisoning. Someone asked me vulnificus. No, it does not. Jo likha hai bas utna swarming you have to know. Coming to the next, falling leaf. We studied this. Girta hua leaf. Girta hua leaf is what? It is going to be Giardia lamblia. Girta hua leaf. So falling leaf is done. All the motilities are done. All these motilities that I taught you were because of flagella. But then I taught you Neisseria and Pseudomonas can be motile because of type 4 pili, and Belantidium coli can be motile because of cilia. Are we settled with this? Now I move on to the next question. Everyone knows it, but it's my duty to make you read this question once before every exam. There is a blind man living in a rural area admitted with respiratory infection, no improvement despite treatment. Patient started on showing septicemia symptoms. Radiology shows a wide mediastinum and Bacillus anthracis along with some spores were isolated from the blood culture. Itna sa padne ke baad they said which phase shows you sporulation, and this is the most gisa pitta. You've seen this in every Q bank, every GT, every subject wise test, and you know it is going to come up in the exam also. So you have to quickly revise with me the entire growth curve. In the growth curve, first question can be label the growth curve. So you should know what is happening in the growth curve. What you see on the x-axis is the time, and what you see on the y-axis is going to be the number of the bacteria. After that, you are going to see a growth curve where the first phase is, ma'am, everything has come to a lag. जो आज यूट्यूब के वॉइस में हो रहा है, लैग. सो लैग फेज इज मीनिंग दैट इज द नंबर ऑफ द बैक्टीरिया इंक्रीजिंग? नो, द नंबर ऑफ द बैक्टीरिया इज रिमेनिंग द सेम. नंबर इज रिमेनिंग द सेम. व्हाट इज इंक्रीजिंग इज द साइज. बेसिकली बैक्टीरिया इज सिटिंग एट वन प्लेस, eating food, going fat, because the bacteria knows that okay, from tomorrow I have to, if you know that okay, from tomorrow my internship is starting, my residency starting, बहुत काम कराएंगे, 36 hours काम कराएंगे, so today let me sit at one place, sleep, eat, chill, relax, and that is what the bacteria is doing. Maximum size is present at the end of which phase? Lag phase. What is it doing? It is sitting and eating a lot of metabolites. It is accumulating because it knows that from tomorrow I have to do a lot of work, and that phase is known as the log phase. Log is what? Number. Ekdam se exponential number increases. So ma'am, just imagine that we were sitting, eating, going fat, and then suddenly we entered into internship. इतना काम कराया कि weight loss हो गया. So can I say that in this phase, when the bacteria is dividing, because of division the number is increasing but the size goes down? That makes sense. Asking you a question from pharma, you must have read it recently. At what phase, if you give antibiotics to kill a bacteria, will you get maximum response? Maximum antibiotic susceptibility is going to be in the log phase. Basically, when the bacteria is dividing, that is when if you start targeting a bacteria with an antibiotic, it will show you the best response. I hope that is okay. So this was my question. When does antibiotic act the best? When does it kill the best? It is log phase. But when is it produced? And that is the stationary phase. You can see after this what happens. The bacteria come to a standstill. Thak gaye. They have now come to a standstill. So in stationary phase, the way you write stationary, I'm just going to convert it, and 4 things happen in stationary phase. Number 1, the bacteria decides let's rest. That is going to be sporulation. Second is going to be toxin, exotoxin production. Third is antibiotic production, and fourth is bacteriocin production. So ma'am, it has now decided to sit at one place and produce a lot of things like toxins, antibiotics, bacteriocin, spores, and that is the question that came. Spore formation occurs in which phase? Stationary phase, and that finally says relaxation kara to dead. This is exactly what happens in your GTs also. First, your GT score doesn't increase, then your score starts increasing, but then you hit that most dreaded plateau. Now, it is here that you decide that if you stayed consistent, there will come a time when you will rise again. But if you give up, is then when the downfall starts. So, your GT and bacterial growth curve is so similar to each other, because finally comes the downfall, that is known as the death or the decline phase. Is that okay with everyone? Yes, all said and done. Now can I move forward to an INI-CET previous year question? But FMG students, don't run away, because this will come in the exam for you also. Because this is the era of resistance. 86-year-old male with a known case of diabetes 3 years comes to the emergency room complaining of a non-healing foot ulcer, which every diabetes patient is going to come to you in the exam with. Patient complained taking different antibiotics for the foot ulcer, not showing response. So this means there is some drug resistance. Which of the following statements is false regarding the drug resistance? Yes. Yes, guys, what is the answer over here? Okay. B, everyone is saying this is what you are suspecting. Let us start reading. What I'll do, I'll teach you whatever is written over here, and then I will come back to this question. Basically, when they say drug resistance, for pharma I'm teaching you, there are two drug resistances possible. By the way, that reminds me, many of you had told me that, ma'am, can we get checklists and can we get flashcards for all the antibiotics, antiparasitic, antifungal drugs at one place? So today's bonus slides are going to be that only. At the end of today's PDF, you will find a flashcard of all antifungal drugs with organism, antiparasitic drugs with organism, which I have added as today's bonus slides. Now coming back, the resistance part, of course, I'm teaching you. There are two resistances. What you study very commonly is mutational drug resistance. Let's give you the example of TB. Now, when you're talking about TB, let's think of what? Rifampicin. Now, ma'am, we have studied that rifampicin का तो resistance होने लग गया है ना? तो resistance क्यों होने लग गया है? Because TB has started showing which gene mutation? RPOB gene mutation. So that is mutation. How many? One mutation. So I call it mutational drug resistance. So ma'am, this is going to be very easy to treat. Why? Because then I know now one drug is resistant, alternative de do. That's about it. So it is very easy to treat. I know kacha one resistance, replace the drug, story will be settled. On the other hand, when I talk about transferable drug resistance, that means that we always used to say na, in school, if there is one naughty student in the class, he will influence all the other 40-50 students, and then everyone will want to be like him. So this is that one bacteria which says I am resistant, I won't get killed by antibiotics, and then he transfers this to every other bacteria and makes every other bacteria a naughty student. So what is transferred? Ma'am, that transfer is going to be of RTF. So this big circle over here is RTF, that is resistance transfer factor. This is like a big taxi. And this taxi is carrying these chota antibiotics. Chota people, it has passengers, it has made to sit in it. These small passengers are known as resistant determinants. So can I say, for an example, this one taxi is able to transfer 1, 2, 3, 4, 5, 6 drug resistances in one go. If any bacteria gets this, uska to, ma'am, it's a jackpot. In one go, that bacteria becomes resistant to 6 antibiotics. So this is very dangerous, and may God save this person. This is very difficult to treat. This means this is the bacteria's strength, power. If he or she, bacteria, gets it, then it has won a jackpot in life. Let me come back to the question. Let's read options. In mutational drug resistance, one drug resistance is seen at a time, like in rifampicin. That is correct. So if I use a drug combination, I can prevent it. Like instead of rifampicin, I give another drug. I can easily prevent it by drug combination, correct? With transferable drug resistance, is it a more severe resistance? If it is transferred 6 in one go, yes ma'am, it is more difficult. Virulence is decreased in transferable drug resistance? Not at all. Ye to bacteria ka strength hai. Virulence will be increased in transferable drug resistance, because that is the jackpot that the bacteria has won. This was the false statement, and that is the answer over here. I think this is okay with everyone. But now a question coming is कि ma'am, mutation में तो mutation हुआ था, transfer में this is getting transferred, but how? How is the bacteria transferring this to another bacteria? What is that going to be? That is going to be the plasmid. Okay, someone said, ma'am, don't annotate the diagram. फिर कैसे करें? How will I teach you if I don't annotate the diagram? I will have to, but I don't think that should become much of a problem for you guys. No, that is not by transformation. If I say that one bacteria is going to transfer it to another bacteria, plasmid, that is conjugation, and that is what I'll teach you, but I will teach you when you answer one question. The cell in which the F factor carries along with it some chromosomal genes are known as. You give me one question, and then Yes, guys? It is going to be F plus is what everyone is saying. F plus, are you sure? No, C, F prime. Okay, so we'll study this. What I'll do, I have got three possibilities and then I've got three different reactions that is going to show. So, pura half an hour of genetics I have put into one slide. In 5 minutes, we have to revise. What are you going to do? Number one, start labeling. Over here, we have a bacteria, ma'am. ये तो bacteria का DNA हमें पता है. And second one is the fertility factor. Every bacteria that has the fertility factor is known as F plus, that is a male bacteria, and this one does not have the fertility factor, so this is F minus, that is the female bacteria. Now just imagine if there is a connection between these two. This is what you were calling as conjugation tube, and now what will happen is this fertility factor will just go and sit over here. I have two options for you. यहाँ से cut होके paste हुआ, control X control V. So is it cut and paste that has happened, or is it copy and paste that has happened? Ma'am, copy and paste. This will not go गायब, this will stay over here. It makes कॉपी करा एंड कॉपी करके यहाँ पर एक पेस्ट कर दिया. सो देर इज अनदर फर्टिलिटी फैक्टर दैट इट विल कम एंड सिट ओवर हियर. कैन आई से व्हेन देर इज अ मेल इन द बिगिनिंग एंड द फीमेल इन द बिगिनिंग, F plus F minus, and conjugation occurs between both of them, in the end what is going to happen, both of them will end up becoming F plus F plus. This we studied. Now instead of F तो fertility था मैम, क्यों बात कर रहे हैं? You were teaching us resistance. Instead of F, if I write R, it's the same thing. Now I'm transferring what? Now I'm transferring resistance. If they write F, they are transferring fertility. If they are writing R, I'm transferring resistance. Now let's go on to situation number 2. Situation number 2 tells me that this time the same DNA was there, the same fertility factor was there, but both of them decided कि हम आपस में we are going to combine. What kind of a male is this? This is a recombinant male known as HFR male. This is known as HFR male because both of them recombination हो गया ना, both of them got recombined. Now imagine this wants to go and have a conjugation with a female, F negative. Do you think anyone in a relationship is allowed solo travel? No ma'am, not allowed solo travel. They will have to travel साथ as a couple. So if they have entered, till the time they were solo, they were allowed to travel. Now they are a couple, so they will have to travel together. So imagine the fertility factor has started traveling and then the DNA has also started traveling, but midway this path breaks, the bridge breaks. Now what happens? Little fertility factor has reached over here, little DNA has reached over here, but ultimately nothing has reached finally. So will the F minus convert into F plus? No ma'am, the F minus remains F minus. Why? Because midway the tube breaks. It's something like whenever there is a relationship, a fight has to happen. So if they entered into a relationship, they also had a fight and then the tube broke, as a result of which no one can reach the final destination. So they will ask you, if there is an HFR male and that undergoes a combination with a female, in the end the female remains F negative only because the tube broke down. Now coming to the third situation, what is going to happen over here? Ma'am, they were having so many fights that they decided that we are now going to separate. Separation is best for us. So once again I will notice that the DNA has become separate and the F has become separate once again. But now remember, now even if there is a separation between them, everyone takes their gifts. I mean, if you've got an iPhone, you will take it with you after the breakup. That is what it believes in. So it says I will take a little bit of DNA away with me, and that is what is known as F prime. This is known as F prime, written as that F dash. Can I say it is the F plasmid which has carried the chromosomal genes and it has taken them away? Imagine if now this decides I have to move on in life, I have to go and conjugate with a female. Now can it copy and paste again? Yes. Can it now go to the female, and can this female now become F prime? So can I say if there's an F prime conjugating with a female, now both of them will become F prime? F prime. So simple way of learning is that if the male is F plus, female becomes F plus. If the male is F prime, female becomes F prime. The only time female doesn't change is when the tube broke off in this recombination situation. If I come back and ask you the question, the cell in which the F factor carries along with it some chromosomal gene, breakup ho gaya, F factor says I will now carry the DNA with me. What is that known as? That is known as F prime. Answer one more question for me guys, a new question. There's a bacterial cell where the F plasmid is integrated with the chromosome. F plasmid is integrated. Integrated matlab relationship enter. What is relationship enter known as? It is high frequency recombinant. Excellent. We are done with this question. Now I take you to a question which is from your previous years and something very lengthy but very simple. There is a 50-year-old male patient presenting to the outpatient department with hypotension, shortness of breath, swollen feet and fatigue. Seems to be somewhat in a case of shock, known case of ischemic heart disease, and had a history of a snake bite 6 hours back. CPC was done, they are discussing this, they have decided that the microbiologist says that I want to sterilize certain heat sensitive liquids. How the question started versus ending is like so different. You thought you were in forensic, then you thought in medicine, you finally come down to microbiome. If you have heat sensitive liquids, rule is don't apply heat because they are sensitive to heat. So how are you going to sterilize this particular situation? So ma'am, dry heat uses heat, autoclave uses heat, pasteurization of milk uses heat. The only thing that doesn't use heat is filtration, like filtering air and filtering water. That doesn't use heat. So for heat sensitive liquids you had to apply common sense. But now you will tell me all the others. Firstly, give me an example of dry heat. One oven that you always get a question on is known as the hot air oven. You will also get a photo of the hot air oven. I think it looks like an oven and everyone knows. What is the temperature? You will tell me. Fill in the blanks. Now starting 10 minutes of only temperatures. What is the temperature? 160 degrees Celsius into 2 hours. You see a lot of variations in many sources. This is what is the most standard protocol. Some places you will see 170 for 1 hour, 180 degree for half an hour. वो सब variations है. Standard is 160 degree 2 hours. What you keep inside it? Number one, any tube, any flask, मतलब anything to do with glassware is kept inside it. Next, you must have seen in the labs, ट्यूब के अंदर एक कॉटन लगा होता है so that the liquid doesn't evaporate. So cotton swabs can also be sterilized in a hot air oven. Very good. And everyone started singing one song, greasy, oily, liquidy, dusty. What is that song? Any greasy material can be sterilized. Any kind of greasy or oily material can be sterilized. Then any dusty material like dusting powder can be sterilized. And liquidy means liquid paraffin can also be sterilized. So repeating, greasy, oily, dusty, liquidy, ये सब चीजें go into hot air oven. So hot air oven is done. So this is great. Now I come to what are these two? That was dry heat. Ma'am, only heating and that's it. Nothing more. But when I talk about the other two like autoclave and pasteurization, they are moist heat. Means you are creating vapors. Like in a pressure cooker, when you're cooking rice, you are creating those whistles that go up. Those are actually creating steam. You're creating moist heat, vapors. That is what is moist heat sterilization. But before I start with them, you will first tell me the wrong combination. Then I will start teaching you. Let's get going. The wrong combination mentioned over here: tyndallization, inspissation, Holder method of pasteurization, or autoclave. Okay, everyone's told me autoclave, wo to rata hua hai na, that I'm not asking you only. Most of you have said option C is going to be incorrect. Okay, I will come back to this. Let me teach you. When I'm teaching you moist heat, you have to go. The season ongoing right now, IPL and batting. That is what we are wanting to see, but we are studying for the exam, so ma'am, we are seeing in breaks. Never mind. We will make an own version of IPL and bat for ourselves. On moist heat, firstly, this is better than dry heat. Why? Because you have created vapor. Vapor will provide more heat later on. So this is a better method. Next, for below 100 degree, at 100 degree and above 100 degree. So for below 100 degree, we have kept IPL. IPL means that at below 100 degree, you have two methods, that is inspissation and pasteurization. Pasteurization obviously ma'am is done for milk. Hamari country mein what is happening? We have Holder method, flash method. So abhi you are seeing only highlights of IPL. Okay, today I am also showing you only highlights of microbiology. Okay, so now sad life. Holder method. Holder method, ma'am, hold karke rakhna. 6330, everyone said. Very good. 6330 means 63 degrees. Hold karke rakhna for 30 minutes. Flash is like flash, seconds. 7220 means higher temperature, but jaldi se, seconds, 20 seconds. 6330, 7220 minutes and seconds you will apply based on Holder or flash, which is said to be better. Ma'am, flash is said to be better. Why? And in our country we prefer flash because flash can kill something which no one else can kill, and which is that bacteria which causes, or I've written the bacteria, it kills Coxiella, ma'am. That is why it is better. You will tell me which fever does Coxiella cause. दो question बताओ, Coxiella का fever and Coxiella का वो fever से granuloma of pathology that I have taught you. Most of you are saying, ma'am, Coxiella causes Q fever. So which granuloma is seen in Q fever? Do you see Dirk granuloma, stellate granuloma, donut granuloma? We always used to make a donut and a Q out of it. So in pathology bonus slides, I hope you saw in the PDF also, I had written Q fever causing donut granuloma. Very good. So Holder method to ho gaya. Flash is better because it kills Coxiella. After pasteurization, we come to inspisation. Now inspisation is a 3-day procedure. Day 1, day 2, day 3, and below 100 rakhna hai na, so you do 80-20, 80-20, 80-20, which means 80 degrees for 20 minutes, 80 degrees for 20 minutes, 3 days you go ahead. Now exam mein, please, chota sa request, 20 ka 25-30 minutes kar diya to timing thoda upar niche chalega. Like in the previous question, for inspisation, they had written 80-20, 80-20 ki jagah they wrote 80-30. Why did I give it a benefit of doubt? I said, chalo, let's consider this because I can see the wrong combination. In Holder method, they have written 7-2. No, 72 degree is going to be for flash method. Holder method, to ma'am 6-3 degree tha, 30 minutes tha. So this is totally wrong. Hence, I selected this answer. So you will give it the benefit of doubt. But mostly it is 80-20, 80-20. Where do you use inspisation for? I use it for two things, LJ and LSS. I am not writing this. LJ media, to hamari country mein reh ke you don't know LJ media is for TB, means you are living under a rock. Everyone knows. You might feel such a stupid question, ma'am, you're teaching us. But do you know in November INI-CET, they asked you such a simple, straightforward question. I myself have read that question 4 times over when the students told me that kuch to catch hoga, kuch to there is a twist in the story. But it was very simple. LJ media is used for the diagnosis of tuberculosis. How is that going to be cured? How is that sterilized? By inspisation. Next is Loeffler serum slope. So Loeffler serum slope is what? It is again for diphtheria, which I will teach you tomorrow, inspisation. So inspisation for two things. One is going to be LJ media, one is going to be LSS media. Coming to at 100. At 100, why? Both things at 100. Boiling is also at 100, tyndallization also at 100. But the thing is that the boiling does, you boil water, is that safe? It is not safe. You boil for 10, 15, 20 minutes, that will not kill the spores. So then you said if you want to kill the spores, you will have to do the same thing 3 days over. So now you will do the same 100 degree wala procedure, but for 3 days. So now it becomes 120, 120, 120. Can you all remember? Inspisation was 80-20, 80-20, 80-20, and tyndallization becomes 120, 120, 120, and 3 days karoge, to ma'am spores will be killed. Boiling to ek 10-15 minute ka tha, boiling water, that did not kill the spores. Tyndallization had killed the spores. Okay with everyone? Coming to the last one, above 100 autoclave, I'm not even asking, you guys have written it so many times. 121 degree for 15 minutes. Pressure kya rakha tha? Like a pressure cooker, 15 psi. This pressure cooker, long wala pressure cooker, is an autoclave over here. This question no one will ask you. They will ask you something else. If you want to kill prions ke liye what will you do? Ma'am, I will have to increase the temperature also, and 15 minutes mein nahi chalega. You will have to put a dot and you will have to convert it into hours. So firstly, this is 134 degree for 1.5 hours. That also I think I will be able to kill prions. Prions are the toughest to kill in sterilization. Question number 1. Question 2, what all do you, where do you find autoclave? I find in the OT, because all the sutures have to be autoclaved. Now you just do fill in the blanks, guys. All sutures. Before an OT, what all you need? Ma'am, we need aprons. They are put inside autoclave. We need instruments. The surgeon needs. They are cleaned by autoclave. We are going to need all the sutures. They are cleaned by autoclave, except catgut. Catgut says, I will not be able to survive 121 degree. So no. Next, all culture media. In microbiolab, lab, all culture media, blood agar, chocolate agar, etc., are cleaned by autoclave, except two. What are the two? LJ and LSS. Why is it that LJ and LSS are kept at such a low temperature, below 100, and all other are able to be going at 121? Because, ma'am, LJ and LSS contain egg. Now, anything containing egg, you will have to keep under 100. If you keep it at 121 degree, it will become anda fry. It will become an omelette. So, you can't keep egg wala things at 121. You will have to keep it at a low temperature. Okay. Finally, please remember all media except LJLSS, that is except LJLSS, and all sutures except catgut. Question yahi par aayega. Okay, now coming back, Dr. Abhinav, you are saying sharps. So if you are asking about sharp, let's say scissors, or you say sharp, let's say a scalpel blade. So what is happening is, can it be done in an autoclave? It can be done in an autoclave, but they have separate pouches. Like probably if you're keeping a culture media, I'm not going to throw the sharp scissors along with it. So now the latest guidelines say that sharps can also be put in autoclave, but there are separate bags that come, you put, which are of course permeable so that the heat can penetrate. Sharps are put in that. Let's assume you want to put a scissors. So now the rule is that you're not going to put the scissors in the autoclave like that. There is a separate pouch, and you will keep the scissors always in open state so that every surface is exposed. Don't close the scissors and put it inside the autoclave. Open it, put it in a separate pouch, and then put it in the autoclave. Now coming back, I have studied all of these things that you had to. सबके temperatures हो गए. Going to this question also, tyndallization ठीक है मैम 120 इंस्पिसेशन 80-20 का 30 कर दिया चलेगा. ऑटोक्लेव 120-115 डिग्री चलेगा. द रॉन्ग वन ऑल्सो वी हैव सिलेक्टेड, ये भी हो गया. नाउ मूविंग ऑन टू द नेक्स्ट क्वेश्चन विच अगेन इज अ वेरी 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 इंपॉर्टेंट क्वेश्चन. बट हाँ, बिफोर आई मूव ऑन, नो आई नो यू विल स्टार्ट आंसरिंग. बिफोर आई मूव ऑन, completing the previous question, sharps are, can it be put in autoclave? कैन इट बी क्वेश्चन है तो कैन तो आई हैव टोल्ड यू डिटेल्ड, यस सिजर्स को खोल के अलग पाउच में. बट इज इट लाइक द मोस्ट आइडियल मेथड ऑफ पुटिंग शार्प्स इन ऑटोक्लेव? इफ आई आस्क यू, सो आई कैन सी, is it the most ideal method? Don't answer that question, answer my question first. So now, yes, is it the ideal method? Ma'am, nowadays they are saying yes, you can put it even in routine practice, you can, but textbooks abhi bhi kehte hain that hot air oven is preferred. But I would not expect that you would get a question on sharps, because to be very honest and telling you what is happening practically, autoclave may, even in the OTs, the sharps are being put. We have seen this in practice, but textbooks say that hot air oven is better for the sharps, etc. So I don't think that kind of a question would be put up, because what is being practiced globally is not something that they would टेस्ट यू ऑन. कमिंग टू द नेक्स्ट, सबने ऑलरेडी आंसर कर दिया था. पढ़ा ही नहीं मैम क्वेश्चन, लास्ट लाइन पढ़ी, व्हाट इज यूज्ड टू डिसइंफेक्ट द ब्रोंकोस्कोप. बट देन दे हैड अ फुल ईएनटी हिस्ट्री, नॉट यूजफुल. ब्रोंकोस्कोप, सारे स्कोप्स के लिए साइडेक्स. व्हाट इज साइडेक्स? साइडेक्स इज गोइंग टू बी ग्लूटाराल्डिहाइड. सो नाउ द रूल इज दैट फॉर एवरी स्कोप you are going to use glutaraldehyde. Glutaraldehyde in the market comes by the name of Sidex, whether it is a bronchoscope, whether it is endoscope, etc. scope. But what if I say your scope, that is the one you wear in your neck, the stethoscope? वो तो patient वाला नहीं है ना, so don't mess up there. For stethoscope cleaning, you don't go ahead with Sidex. For stethoscope cleaning, you are going to use isopropyl alcohol. Don't mess up there, first exception. Second, coming back to the patient wala scopes, if they write, very good, I got that ma'am, ureteroscope and arthroscope. Yes, for ureteroscope and arthroscope, what is preferred? Plasma sterilization. So now listen to the three questions. All the scopes, bronchoscope, endoscope, etc., go by Sidex, that is glutaraldehyde. But all the ureteroscopes and arthroscopes are going to go where? They are going to go by plasma sterilization. But apna stethoscope mat usme add karna, that is cleaned by, which no one cleans. Two things which no one cleans is your mobile phone and your stethoscope. You go into the hospital with it, you come back with it, you eat your food with it, and then you are spreading the infection to yourself. Learn to clean your stethoscope. How? By isopropyl alcohol. One more thing I see over here is, ठीक है, alcohol is okay, glutaraldehyde, formaldehyde is okay. What is this written? 1% sodium hypochlorite. When do I use sodium hypochlorite? वो क्या घिसा पिटा PSM का question आता है. In case of what situation am I going to use? In case of blood spill, you are an intern, you have thrown one vacutainer of blood on the floor. Now which sterilizing material will you use? You know that question, that is for blood spill. But coming back, you said everything is by Sidex, but arthroscope and ureteroscope are by plasma sterilization. So can you tell me this particular question? All are true about Sterad except, my first question is, Sterad is the name of this machine. Obviously it's for sterilization, but they will ask you for what sterilization? It's the machine for plasma sterilization. So that is plasma sterilization. So now tell me, what is plasma? What is this? First answer the question. All are true except products are less toxic than ETO, uses Bacillus stereothermophilus, can be utilized for products which are heat sensitive, and chamber carrying out plasma sterilization uses ambient air. Everyone says ma'am option D incorrect है, क्योंकि plasma को रखना है तो फिर ambient air में नहीं होगा, it is going to be via vacuum. So let me tell you what is happening in plasma sterilization, then you will answer the question once again. So this is that dabba, that white color dabba called sterad for plasma sterilization. And in plasma sterilization, number one, the temperature is always kept very low and the medium is always kept as vacuum. So khud socho, anything that is heat sensitive, I know I'm keeping a temperature of, you don't have to learn it, but temperature is like 50 degrees, काफी कम होता है. So anything that is heat sensitive will easily be able to, I can clean it over here. Now what are the chemicals used? How have you created plasma? Guys, this is not the plasma, that blood wala plasma. This is the fourth state of matter. You have created plasma. How have you created plasma? By hydrogen peroxide. So we know that, oh, hydrogen peroxide, to ma'am, wo free radical hai. It is anyway very, very dangerous. From that you have ended up making plasma. That plasma will also be very, very dangerous. Now tell me, what are the uses? I think you have already mentioned the arthroscopes and the ureteroscopes are the two uses that we are going to have over here, which we already know. What is the control? Bacillus sterothermophilus, which means the next question is, you will tell me all the controls for everything that we have studied. But before that, coming to this, do you know you have done something? Everything you have kept very, imagine being very, very low key and low profile in life and attaining the best results, and that is what you have done in plasma sterilization. That is your take home message from today's class. You have to be like a sterad machine while you're studying. You have to be very, very low profile, very, very basic in your approach, and then aim for the highest result, which means it decided, I'm not going to boast a temperature of 121 like an autoclave and a pressure cooker and make those noises of a whistle. I'm going to keep my temperature very, very low. I'm going to work in a closed situation with vacuum around me. I'm going to use H2O2, but then I am now going to kill everything that comes my way, every bacteria, every parasite, every virus. So that means that plasma sterilization is going to be the sterad system. Very good. Coming back, will it produce any toxic products? No. Thoda बहुत toxic products everyone produces, but it is, like I said, it is very, very low profile, but it has made the best result. It has very less toxic products. If you compare it with others, whom are you comparing it with? Let's compare it vis-a-vis ETO. What is ETO? ETO is ethylene oxide. Ma'am, ethylene oxide is a method which I use for, again, for sterilization, but this is a gas, and this gas has so many toxic products. I mean, it can cause cancers, it can cause fires in the lab. So it is not that boisterous. It is not that plasma sterilization will do all of that. It is a very low profile, no toxic products. So start comparing. Number one, does it produce less toxic products like ethylene oxide? Yes. Is it using Bacillus sterothermophilus as control? Can it be used for heat sensitive material because it is low temperature? Yes. Chamber uses ambient air? No, it uses vacuum. So I found my wrong statement. I think this is clear. Now we come to where is acha. Next question is, plasma sterilization uses what? H2O2. Ma'am, you were comparing it with ethylene oxide. Can you tell us ये ethylene oxide जो gas है वो use कहाँ पे होता है? It is used in all the cardiopulmonary machines. So all the ventilators, advanced machines, cardiopulmonary machines are actually going to be cleaned using gas sterilization. Okay, Dr. Ashmit, we are done with this. Now you will tell me control की बात शुरू हुई ही है. So for this you will tell me which organism is commonly used as the biological control for this gas sterilization. Very, very danger, can cause fires, can cause cancers, but useful भी है. Ethylene oxide का control क्या होगा? Ma'am, G for G, gas का control GloBG, gas का control GloBG. Now what happened in the exam? GloBG was not there in the options and students kept searching. Gas के लिए GloBG ma'am ने पढ़ाया था. Do you know GloBG is the new name? What is the old name of Bacillus GloBG? It is subtilis, and then you had to mark Bacillus subtilis, whatever may be there. This came in the FMGE exam last time, and I felt so sorry for students because वो याद करके गए थे G for G, G for G. They kept searching GloBG मिलेगा, GloBG मिला ही नहीं because examiner gave the 20 years पुराना नाम, and examiner gave Bacillus subtilis. That is the same thing, but then why should we learn 20 years पुराना नाम? I don't have an answer to why you should learn it, but it has come as a question. Now I will make you learn it. Now I have got all the controls that you have, and that will finish off this particular situation. When I have these controls, you start from gas sterilization only. Ma'am, you said G for G. So इसके लिए क्या होगा? There is Bacillus GloBG, but like you said, ma'am, Bacillus GloBG नहीं होगा. We will mark Bacillus subtilis. In fact, when in doubt, I'm saving you, I'm giving you a kind of a jugaad over here. When in doubt, mark Bacillus subtilis. For hot air oven, what is the control? It is again Bacillus subtilis, uska cousin brother Bacillus atrophius. If it is not there in the options, then Clostridium tetani. Are you using these bacteria as the controls? No, ma'am. These are the biological controls. Biological matlab I am using their spores. These are all biological controls and I am using the spores of these bacteria. That is why I am again and again saying bacillus, bacillus, because bacillus wala bacteria makes spores, right? Okay, subtilis also, atrophius also, and tetni for hot air oven. Then comes the two. ऑटोक्लेव एंड प्लाज्मा स्टेरिलाइजेशन, एक है हाई टेम्परेचर वाला 121 डिग्री, एक है लो टेम्परेचर वाला, बट दोनों का कंट्रोल इज द सेम, दैट इज बैसिलस स्टेरोथर्मोफिलस. कमिंग टू द नेक्स्ट वन, रेडिएशन. रेडिएशन के साथ कौन सा अल्फाबेट जाता है? विच वन विल यू मार्क? मैम पी गोज विद आर, सो बैसिलस प्यूमिलिस इज गोइंग टू बी फॉर रेडिएशन. कमिंग टू फिल्ट्रेशन, B diminuta. Here B is not bacillus, B is Brevendimonas, but you don't have to learn Brevendimonas. You have to learn diminuta, minute, filter. Filter का pore size कितना होता है? मैम filter का pore size is question. Filter minute सा pore size 0.22 micron, so minute सा bacteria, that is diminuta. Diminuta. If diminuta for INI-CET students, if minuta is not there, then which other bacteria is very small and can be used over here? Then small wala bacteria is going to be Serratia. Is this okay with everyone? Yes, is going to be Serratia. So या तो minuta वाला minute होगा या small वाला Serratia. Someone asked me that ma'am, are these all killed? Just tell me, if they were anyway killed, then what are you trying to test? They have to be alive. The point is that you are, let's say that you have put Bacillus stereothermophilus in an autoclave. Why have you put it? Because you want to find out that is autoclave working properly? Will autoclave be able to kill it? So you have to put the spore and then you have to wait that has it been killed. If you put a killed organism pehle se, then what will you test? So you can't put a killed organism inside it. One thing is done. What did I mention? Ma'am, I'm using biological controls. One question comes that you don't want to deal with spores because you feel that people around you will get infected. आपको ऐसा OCD है कि मुझे ये biological spores नहीं use करना. Can you use some chemical method and name a tube which can be put? Chemical method for control is known as the Brownie's tube. You will not get the principle. It is a tube which has a chemical in it, and what do they do? Let's assume I took that tube and it had a reddish color chemical in it, just giving you as an example, okay? I took this tube and I put it inside a hot air oven. What is the temperature for hot air oven? You said 160 degree for 2 hours, na? After 160 degree for 2 hours, I saw that this particular tube changed its color to green. And then I'm like, okay, फिर तो काम कर गया. If the chemical has changed the color, this means the hot air oven has worked, autoclave has worked. So if you use chemical, what is that tube? Brownie's tube. What do you use for biological control? These are the spores, and that finishes it over here. Now coming back to cell wall. Let us see. Let us come back to cell wall. Which of the following subsequent constituents of fungal cellular wall is absent in cases of mucormycosis? Cell wall तो है, but bacteria का नहीं. अब fungus का cell wall है. I've given you a random diagram, but you will not need it. What is absent? What is absent over here? You are saying B, beta 1,3 D-glucan. Yes, and I think पूरे fungus, मतलब I'm starting with mycology, पूरे fungus chapter में, now my aim is that I should do the fungus chapter approximately in 30 minutes, 30-35 minutes of fungus. What we will do, I'm teaching you the most expected question first before you sleep off. So what is it that I'm talking about? Do you know beta 1,3 D-glucan? कोई mark नहीं कराएगा, बस इसको एक बार देख लो. This is the fungal cell wall. I can see 2-3 very important things in fungal cell wall. Ma'am, यहाँ पर वो नैम नैग वाला स्टोरी नहीं है. नैम नैग वाला स्टोरी वाज इन बैक्टीरिया. हियर व्हाट आई नोटिस, नंबर 1, I can see fungal cell wall has chitin. What will you learn? What does the fungal cell wall has? It has chitin, and secondly it has beta 1,3 D glucan जैसा structures. बस ये दो याद रख लो. One is chitin and the second is going to be beta 1,3 D glucan. But now they are saying that which of the following constituents are going to be absent in mucormycosis? This means beta 1,3 D glucan is not in mucormycosis. Repeating once again, beta 1,3 D glucan testing अगर करा तो मैम you will say that all fungi are going to have it, except Bangalore Medical College. What is Bangalore Medical College? B for blastomyces does not have it, M for mucor does not have it, and C for cryptococcus does not have it. Repeating, blastomyces, mucor and cryptococcus, they are not going to have beta 1,3 D glucan. Please note, this is, I mean, how many times should I say it? The most expected question. It does not have beta 1,3 D glucan. So now can I say that if you are doing beta 1,3 D glucan testing and it comes out to be negative, someone gave you a hair or a nail or a skin sample, beta 1,3 D glucan test came out to be negative, you will say I have only 3 possibilities left: blastomyces, mucor and cryptococcus. इन्हीं तीनों में से something could be there. There is one more thing that is there in the cell walls. Very good. Someone said galactomannan. Okay, so galactomannan test is again for all fungi. Galactomannan test is positive in all fungi, but when galactomannan test comes positive, I always think of mostly invasive aspergillosis. I'm giving you one rule over here that whenever they say galactomannan test is positive, you will say that, ma'am, I could have thought of every fungus and they were all fungal organisms in the options. Single best answer क्या select करके आओगे? Invasive aspergillosis. Yes, this means कि mucor में even galactomannan is absent. It is going to be absent in mucor. So mucor, remember, was that famous black fungus. COVID वाला black fungus was mucor. So imagine COVID में, when we were anyway falling short of tests, this black fungus mucor decided कि मैं सब test negative दूंगा और ज़्यादा these people will be troubled because of me. Neither will I come positive on beta-1,3-D glucan, neither will I come positive on galactomannan. Now the microbiologist can sit and cry all night because I am not giving any test positive only. Imagine the situation that must have arisen at that time. So it was an altogether different thing. But coming back, mucormycosis does not have beta-1,3-D glucan, mucormycosis does not have galactomannan. If beta-1,3-D glucan is positive, you can think of any fungus except BMC. If galactomannan is positive, you can think of any fungus, but most common answer, invasive aspergillosis. Now coming to the next question. There's a patient presented with a scaly skin, redness and irritation and small blisters all over the skin. So skin sample लिया होगा, fungal infection suspect करा. Which culture media will you use for the fungus? They've asked you a very simple question. What is the culture media for fungus? It is SDA. सबके बारे में मुझे एक एक question बता दो. Where do you use PT? I know I'm asking you bacteria today only, but why not? पोटेशियम टेल्यूराइट agar कहाँ यूज करते हैं? पोटेशियम टेल्यूराइट agar is going to be used for Corynebacterium diphtheria. Tomorrow I will teach you. It is the selective media for Corynebacterium diphtheria. Someone had asked me, Dr. Arjit had asked me, rhizopus, mucor and absidia. Dr. Arjit, don't they come under the same family of zygomycetes, right? So we have three things under zygomycetes, rhizopus, ram, we learn it as ram or ram, that is going to be rhizopus, absidia and mucor. So the same family we are talking about, everything goes common for them. Okay, so PTA to nahi hai ma'am, that is for Corynebacterium diphtheria. Where do you use NNN? Today we will study NNN is in parasitology. That is, it is number 1 for Trypanosoma cruzi. Remember the cruise, jhakas like, which I will teach you, just Chagas disease. And for Leishmania, the Kala Azar, that is going to be triple N. So SDA is obviously going to be for fungus. SDA stands for Sabauroud's dextrose agar. Ma'am, photo ka question dikhane se pehle, what are the questions that I get? Number 1 question, what is the pH of Sabauroud's dextrose agar? pH will be asked. Acidic hota hai, 5.6 ke paas. The way you write Sabauroud's, you will remember the pH is somewhere around 5. So it is 5.6. Now coming to what all does it contain? It has gentamicin. What do you think gentamicin must be doing? Ma'am, ये तो bacteria की growth, because fungus grow करना चाहते हो ना, so you don't want the bacteria to grow, so gentamicin will not let the bacteria grow. Cyclohexamide क्या होता है? It inhibits the fungus. Why am I doing this? When I want to grow the fungus, why am I adding cyclohexamide? Because it inhibits the normal fungus. The saprophytic fungus तो you don't want should grow ना. You want the bacteria, the disease-causing pathogenic fungus to grow. So please note, cyclohexamide is added to reduce the saprophytic fungus. Gentamicin is added to remove what? To remove the bacteria. So two things are added, cyclohexamide and gentamicin. Why did I teach you? Because I'm being nice and I have leaked the next question to you, and that is, which of the following features of Sabauroud's dextrose agar makes it particularly useful for culturing dermatophytes? Number 1, is it a neutral pH? Not at all. Is it acidic pH? Yes, pH of 5.6 and addition of cyclohexamide. I think that is the correct answer. Are you giving extra growth factors to support bacterial growth? No, this is for fungal growth. Neutral pH, again ruled out. Simple. Acidic pH, cyclohexamide, that is going to be Sabauroud's. That's a new question that I got, how SDA practical questions will come. So ma'am, SDA दिखता कैसा है? This is SDA in the center. This is how it looks like. pH 5.6, ये हमने पढ़ लिया. But from this SDA, you said, acha, this white fungus is seen. Now I want to look at this white fungus under the microscope. See, there are two things, and listen very carefully. Today, a patient comes to you like this with a skin redness, irritation, blister, skin ka kuch hai. Ma'am, number 1, so you will take a skin sample. You will take scrapings from the skin. When I take scraping from the skin, I can directly see them under the microscope like this. One possibility, you put some colors, you look at it under the microscope. Second possibility is that you put it into an SDA and you let it grow. So sabse pehle I decided I will look at it under the microscope. When I used a black color stain, that is silver. Remember, silver always gives a black color. So Gomori methenamine silver stain will give a black color to fungus. If I wanted to see the fungus to be pink, I decided it is going to be it is no, I read a question. Cyclohexamide होता है. See, chloramphenicol is separate, cyclohexamide is separate. Madhurimides दोनों चीजें होती हैं, but cyclohexamide is what is added to inhibit the saprophytic fungus. Okay, coming back, when I want it to be pink, pink कौन सा? Pass, this we know. But when I want it to be fluorescent, you've studied this. Fluorescent stain for fungus is what? It is calcoflower white. Now I'll ask you a question. Flower तो मतलब fluorescent microscope. Do you know what it binds to? What was in the cell wall? It binds to chitin. अब cell wall से related सारे questions done. If they ask you what is binding to chitin, ma'am, chitin से calcoflower white binds. And what is the story of beta 1,3-D-glucan that you have studied? पूरा cell wall is settled for you. Calco binds to chitin. Flower means fluorescent microscope. ये तीन तो I saw. I took the skin and I saw it under the microscope. Then I put the skin in the SDA and I saw the growth. Now from the growth I want to do the microscopy. Can I say that I am wanting to do culture morphology? Culture में से morphology, culture में से microscopy, that is done by LPCB, lactophenol cotton blue. I have a question over here, common sense question. Why have you added cotton blue? Because ma'am, I want a blue color. Why have you added phenol? Phenol to ma'am, you've been telling us phenol, जैसे Lysol phenol, like you clean your floors, they are disinfectants, so phenol is a disinfectant. Lacto मतलब lactic acid you have added. तो ये तो no one will ask you what other than lactic acid, phenol and cotton blue is added in lactophenol cotton blue, and that is the fourth thing called glycerol. Now you will tell me a question, what is the function of glycerol and lactophenol cotton blue? So lacto plus phenol plus cotton blue तो ठीक है, plus it is glycerol. Ma'am, glycerol is famous for what? For providing water. The sample should not dry. Skin, hair, nail, they will become very dry. So can I say it will prevent the drying of the specimens, or in other words it is providing water? Can I call it hygroscopic? Yes, I can call it hygroscopic. Very, very good. So from SDA I decided to do blue color ka LPCB. Now if I say these are option A, B, C, D, all four are stains for fungus, but I say which is the stain for knowing the culture morphology? Culture ka morphology ke liye what will you say? You will say it is lactophenol cotton blue. Very good, you are done. I'm asking you first three questions are ऐसे कॉन्फिडेंस बूस्टर वाला क्वेश्चन के खुश हो जाओ टाइप. ओके, सो दिस इज वन ऑफ देम. 28-year-old man presented to the OPD with complaints of recent onset rash. Now the patient is otherwise healthy, no allergy, medication, chemical exposure. Physical examination shows hypopigmented scaly patches on both the arms. The culture was done. The microbiologist could not get results on regular SDA. Which is the fungus that cannot be grown on regular SDA? You are saying Tinea versicolor. Can I also call it Malassezia furfur? Or they call it Pityriasis versicolor. Would that be the same? I'm telling you all possibilities. Disease का नाम लिखा तो Pityriasis versicolor और Tinea versicolor. उसके fungus का नाम लिखा, so that is going to be Malassezia furfur. Okay, now coming to all the photos. Ma'am, right now it was written hypopigmented lesion होगा, so this is what I can see on the back. I can see this hypopigmented lesion. Now you took the skin scraping. अच्छा, what I showed you so many colors, but this is a microbiology without a color. Can you tell me what is this? Ma'am, ये क्या है? Colorless. ये कौन सा आपने mount बनाया? This is the KOH mount. This is referred to as the KOH mount. Perfect. If this is perfect, great. क्या दिखा मैम? यहाँ पे डॉट 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 दिखा. These dot 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 are what? These are going to be the छोटा छोटा yeast forms. And then I can see these long forms. What are they? They are known as the hyphae forms. These are the hyphae. So can I say that this is going to be a chota chota matlab meatball and long matlab spaghetti pasta. So this is the meatball and spaghetti and meatball appearance. In case you're vegetarian, then the long becomes banana and the dot 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 becomes grape. So banana grapes appearance. तो KOH क्यों करते हैं? Before I move forward, what is the reason for doing KOH? What is the primary purpose of using potassium hydroxide preparation? Question में से question. We have gone into a loop. How from one you can derive another? Tell me guys, before I go further, why do you use KOH? Ma'am, K for K. KOH is used because you want to digest the keratin. See, you are taking skin sample, hair sample, nail sample. There are so much of keratin. If there's keratin, how will you see the fungus? So first you will have to remove the keratin and then you will be able to see the fungus. There is no other reason of why I'm using KOH mount. So usually we use a 10 to 20% KOH mount depending. Like imagine skin ke liye to 10% is good enough. Nails are very thick, very solid, thick, right? For that I might need a 20% KOH. So that is the concentration in derma that we use. Now coming further, where else do you see spaghetti and meatball? This only INI-CET students will tell me. Gynae में, pap smear में, where do you see the spaghetti and meatball appearance? It is seen in that strawberry cervix wala, trichomonas. So trichomonas to is that chota chota meatball. What is the spaghetti? That is going to be leptothrix. Very good guys, very very good. Now finally coming back, that is seen in Malassezia furfur. तो फिर हमने इसे culture करा. Now the culture I tried to do regular SDA for a fungus, it didn't work. I had to have olive oil. Once I added olive oil, that will tell me this fungus says I love fat. I am lipophilic. You will have to add some olive oil, then only I will grow. And when it started growing, it started showing me fried egg colonies. So fried egg colonies तो मैम पढ़ा था हमने. We studied it na in the last class. Can you quickly tell me patho ka, micro ka, fried egg appearance? Jaldi, I have to written it. You will tell me guys. Number one, I will write it once again for you. It's all overlapped. So we will write it again later on for spaghetti meatball. But fried egg appearance kisme? Brain tumor, oligodendroglioma. Last to last class, same photo we have drawn. Next, hairy cell leukemia ka, bone marrow biopsy, aspirate, bone marrow biopsy. We have done this. टेस्टिकुलर सेमिनोमा एंड ओवेरियन डिसजर्मिनोमा थर्ड. ये तो था हमारा पैथो वाला. माइक्रो में कहाँ पढ़ा था? यू टोल्ड अस मैम एक फंगस होगा दैट इज मैलेसीजिया फर्फर एंड वन देयर इज गोइंग टू बी अ बैक्टीरिया माइकोप्लाज्मा. ये पढ़ के आए हैं. आई डोंट थिंक एनीथिंग न्यू. दिस इज जस्ट वन लास्ट रैपिड रिविजन. अच्छा, मैलेसीजिया फर्फर का ट्रीटमेंट क्या है? लगाने के लिए भी एजोल, खाने के लिए भी एजोल. दैट इज हाउ इट वर्क्स इन केस ऑफ फंगस. ऑल्दो ऑल द एंटी फंगल ड्रग्स आई हैव रिटन एट द एंड, बट खाने के लिए, लगाने के लिए व्हेन आई टॉक अबाउट topical, it is imidazole. When I have systemic, that is also azole. You can also use sulfur ointments. That is what is an extra question that has been asked in dermatology. वैसे तो it is लगाने का टॉपिकल एजोल, खाने का भी एजोल, बट 10% सल्फर ऑइंटमेंट कैन ऑल्सो बी यूज्ड. कमिंग बैक, आई हैव डन ऑल ऑफ दिस एंड व्हाई डिड आई टीच यू? बिकॉज विच इज द फंगस दैट कैन नॉट बी ग्रोन ऑन रेगुलर SDA. मीन्स आई विल सी इफ यू वांट टू डू फ्राइड एग ना, if you want to do anda fry, for anda fry you will need little bit of olive oil na. That is how I have learnt it. This is one thing. Second, a variation of the same question comes like this. The same patient with discrete hypopigmented macules on the upper back, the patient is a laborer, skin scrapings with 10% soy spaghetti meatball appearance. Same question ka, different variation. Answer will be the same. It is tinea versicolor. Perfect. Moving on to सबका favorite question. I said na, पहले दो तीन chill करो, enjoy करो, you will get right answers. Patient made a self-diagnosis of an athlete's foot and began using a product advertised on TV. That is just every patient in India. The condition improved but did not clear, and the patient then decided, I will show to a dermatologist. The skin scraping was sent for culture, including culture for fungus, and that showed a colony which produced abundant microconidia. All of you are writing A and B. Most of you are writing A. Are you sure it's only A and it is not D? Because ट्राइको तो दोनों है. एग्जाम में भी यही कंफ्यूजन होता है. दे डिड दिस लास्ट टाइम ऑल्सो. ट्राइकोफाइटन एंड ट्राइकोस्पोरॉन, they are totally different. You are right over here. Answer is trichophyton, because I am talking about TME, and these three fungus comes under a family which is known as, excellent guys, that is dermatophytes. Now see the word dermato to will tell me कि हाँ fungal, skin का fungal infection. Everything has to be similar to phyte. So if they write epidermophyton, okay, microsporum तो याद रहेगा. ट्राइको में डिफरेंट था मैम. ट्राइकोस्पोरन लिखी है, ट्राइकोफाइटन. सो व्हेन द फैमिली इज फाइट, then everything I will try to incorporate is phyte. Therefore it is trichophyton. Very good. Everyone has started, ma'am. TME, PSC, another internal thing which only we guys know. What is TME-PSC? TME is the three names of the fungus that comes under it, okay? That is Trichophyton, Microsporum and Epidermophyton. When I say PSC, that's the macroconidia ka shape that we are going to have. So the macroconidia is going to be, in first one is pencil shaped, it looks like a pencil. In the second one, it is going to be spindle shaped, so it is looking exactly like a spindle over here. And in the third one, it is a club shaped, so it is looking like a club. Exam में जाके examiner maybe is also viewing this LRR session and knows, अच्छा इन लोगों ने TME PSC याद करा, let me not write PSC. Instead of writing spindle, examiner tries to use a very fancy word. Fusiform appearance is going to be seen. Examiner says cigar shape appearance is seen. You'll be like, अच्छा मिलता किससे है? Cigar looks like what? Cigar looks somewhat like a pencil only, so I will consider it the same. Fusiform looks somewhat like a spindle only, तो match कर लेना थोड़ा kindergarten students की तरह match the shapes करके whatever shape matches your TME PSC mnemonic you go in for that, because examiner has started confusing you this way. उससे ये leak हो चुका है TME PSC. I will, of all the people, let's ban the examiner from the Telegram group. That's the only way he would not get to know. But coming back, what are we talking about? Macroconidia. What about microconidia? Microconidia go in a descending order in TME. So TME for Telegram, for those who are new to our classes, let's also welcome them to TME is the short form for Telegram, and PSC is Preeti Sharma channel. That is how we have been learning it. Okay, self-obsessed people we are. We made mnemonic on ourselves. Now coming to microconidia. Microconidia maximum to minimum. यही तो पूछा था. Question said there is abundant microconidium. Abundant microconidia T मतलब trichophyton. Clinically how will the question come? ये stain का क्या नाम है? Stain का नाम है LPCB. मैम ये तो हमें पता है. What will it affect? When I say trichophyton, tri मतलब सब कुछ. It will affect skin also, hair also, nail also. In fact, ma'am, the name of the family is dermatophyte, तो skin तो common sense है ना. Everyone will affect the skin. Trichophyton will affect everything. Microsporum will affect skin and hair, and epidermophyton will affect skin and nails. There are two ways of learning it. Some people say epidermophyton में nail लगा देंगे, microsporum में we will put hair. Some people also learn जहाँ जहाँ fight आया वहाँ nail आएगा, because they are like violent people. They say we have to fight with nails, scratch with nails. तो epidermophyton, fight with your nails. Then you put nails over there. Microsporum में you don't put nails, you put hair. Trichophyton में you are back to putting nails. So wherever you want to fight, you have to put the nail is how you are going to learn it. Okay, now finally your clinical part is also done. Dermatophytes are over. But now I ask you, ये trichosporin क्या था? What is this? I will ask you. There is something called Trichosporin BHL. What does it cause? Dermatology question. Trichosporin BHL. Yes guys, जल्दी आंसर. डर्मा क्वेश्चन है. डर्मा LRR हो चुका है. पिएड्रा ऐसे थोड़ी चलेगा. व्हाइट पिएड्रा, ब्लैक पिएड्रा. Piedra, so I should have told you. White piedra, black piedra. Very good. So there are two things. This is Trichosporin BHL. We always study BHL sounds like bijli. People who don't understand Hindi, today we are teaching you one word. Bijli is light and light is white. Okay. Trichosporin BHL, bijli is white. So that causes white piedra, versus if I write piedra horte, whenever you cook something or you increase the temperature, वो hot होता है, जल जाता है खाना. खाना gets black and burnt. So piedra horte is going to cause black piedra, and Trichosporin BHL is going to cause white piedra. These are hair related fungal infections. Don't confuse Trichosporin and Trichophyton. One last time and I'm done. Moving on to a question that came, that is important for INI-CET and NEET PG till last to last year, but I think ये अभी तक FMGE पे आया नहीं, so now it's a trio for everyone. Listen carefully. A second year medical student performing some tests in the lab, demonstrator takes a few strands of human hair and places them on a petri dish. Hair is then treated with distilled water, yeast, fungus, and put in an incubator. 2 to 3 weeks, the demonstrator takes out the hair and microscopically, blue stained wet preparation shows wedge-shaped perforation of the hair. इतनी कहानी के बाद I got to know this is a perforation test of the hair. Which organism will give you positive? You said trichophyton. Very good. Can you tell me which trichophyton? That is going to be trichophyton. Mentagrophytes. Very good. So I'll quickly repeat for you what am I doing. I am doing the hair perforation test. तो मैम इसके अंदर hair लेना है, common sense, and because you're doing it for fungus, you will add the fungus on top of it. And what are you trying to see? I'm trying to see कि hair perforate हुआ या नहीं हुआ, that is going to be hair perforation test. So tell me, in the first photo, do you see any perforation of the hair happened? No. So this is hair perforation test negative. This means that is a case of T. rubrum. Versus in the second photo, I can see that yeah, hair is getting those wedge-shaped perforations. So if hair perforation test is positive, that is Trichophyton mentagrophytes. The shaft is eroded. Very good. Exactly, guys. So remember, Trichophyton mentagrophyte is like fight again, violence. This topic is all about violence. When you will fight, you will want to tear the person apart. That is how angry I become, ma'am, when they tell me na exam 2 shifts mein hone wala hai, sections mein hone wala hai, they change it one night before, mera man karta hai to tear it apart. That is what you're wanting to do. So फाड़ने का मन करता है, you just want to tear that examiner apart, right? Mentagro fight is going to फाड़ देगा, hair को फाड़ देगा, and that is going to be done. What is the significance, Dr. Amit? Basically in dermatology we are dealing with a lot of infections which cause clinically the same thing. So matlab both of these can clinically cause hair infections, but now we want to know the difference between them. So which exact organism is causing it? The exact organism, if you want to know, is when you want to do the hair perforation test. Okay. Coming to the next thing, I think this everyone would be able to tell me because ये तो simple derma question है. Which of the following options listed below is a fungal agent that affects the subcutaneous layer of the skin? Subcutaneous, not skin, below the skin, and that many of you have started answering. C, that is Sporothrix. So what are all the subcutaneous fungal organisms that we have? Subcutaneous fungal organisms are number one, mycetoma. I think everyone knows the mycetoma of that foot. Yes. Second one, rhinosporidiosis. Rhino matlab nose. T for chromoblastomycosis and S for sporothrix, and that is exactly what I had to mark over here. So another MRCS, there are only two, I promise. Entire path micro मिलाकर there are only two MRCS mnemonics that we know. एक हमने शुरू में करा था gram poor organism, that was obviously ma'am, those were the bacteria's MRCS. Which bacterias you know of? Mycoplasma, Rickettsia, Chlamydia and spirochetes. वो बैक्टीरिया वाला MRCS था. This is the fungus वाला MRCS which is subcutaneous, known as mycetoma, rhinosporidiosis, chromoblastomycosis and sporothrix. So this is done. So sporothrix is subcutaneous. Let me ask you another question. 24-year-old South Indian patient. हमारे examiner के बहुत सारे bias हैं, which he will ask you again and again. So now I want you to tell me just by noticing this month, भूल जाओ सब कुछ, क्या diagnosis बनाओगे? South India patient, rhinosporidiosis. Why they say that is because examiner has that bias. I'll tell you what. Like tomorrow I will teach you the history of a truck driver. Now when examiner says truck driver, even if he doesn't write, we have to assume that truck driver ka sexual practices are very unsafe. So you will have to assume sexually transmitted disease, truck driver ko syphilis hi hoga. That is how we go ahead. Similarly, as per the examiner, people in South India are forever swimming. These are some biased things which he has come up with. So swimming se related, the disease that he will think of is rhinosporidiosis. Like so many such biased things. Like when there's an African child, examiner always says Burkitt lymphoma. तो ये उसका bias आपको याद करके जाना पड़ेगा, ठीक है? So 24-year-old South Indian patient, rhinosporidiosis is what he's trying to tell you. Now taking baths in ponds presented with local pruritus, coryza, sneezing, rhinorrhea, postnasal drip or nose related things. The patient is admitted and nasal polyps were noted. Again, rhinosporidiosis. Biopsy showed spherules. And what is true about the diagnosis? Most of you have started saying B is true about the diagnosis. Okay, so let me just show you some pictures. When I'm talking about rhinosporidiosis, I think this is the photo that everyone knows, and I'm going to write it all in one and tell you how to study it. Why am I teaching you this? Because हम subcutaneous layer वाला पढ़ रहे हैं, तो I started with rhinosporidiosis. Rhino word will tell me कि अच्छा मैम nasal के अंदर Rhinosporidium seaberry is the name of the fungus. Now split the seaberry. सबसे पहले rhino and berry tells me कि rhino में berry, nose में berry, strawberry, nose के अंदर strawberry polyp will be noted. Next is seaberry. Sea sounds like sea and ocean. So they will always give me a patient who's taking bath in the pond or the ocean or the nadi or the lake or somewhere. So sea. So much so that now they say that this is reclassified into a sea organism. They don't call it a fungus. Now they say this is an aquatic protozoa. Someone said myculic cells are formed and then deleted themselves, which is so right. You wrote it and then you corrected yourself because Rhinosporidium seaberry में myculic cells नहीं दिखते. Tomorrow I will teach you something called rhinoscleroma. वो अलग है. I will come to that in a while. So now when I say कि ठीक है, nose के अंदर strawberry दिखेगा, C मतलब now it is called as aquatic protozoa. Now this will be C मतलब some ocean ponds for South India. Now I see strawberry polyps over here. How will I do the biopsy? I will do the biopsy and I will see the spherules. I can very well see these round spherules, and that is how this will be done. So what is the treatment? The treatment is not antibiotics. The treatment is to remove the polyps by surgery. So that is going to be polypectomy. So ma'am, can't you culture it on SDA because it's a fungus? No. Please remember, this is a non-cultivable organism. I think everyone knows this. There are non-cultivable organism. This is why the biopsy is done and then you get to see the spherules. That is how you do the diagnosis. You don't do diagnosis on SDA. This is a non-cultivable organism. Okay with everyone? Coming back to what someone asked me, I will take a separate slide and answer it. Someone said that ma'am, ye jo rhinoscleroma, I'm writing it in as bold the pen can go. Rhinoscleroma is different. See, you are getting confused because that is going to happen in the nose, but look at scleroma. That is caused by Klebsiella rhinoscleromatis. देखो उसके नाम में है. That is caused by Klebsiella rhinoscleromatis. Rhinoscleromatis. Please remember this. So rhino for nose and scleromatis for scleroma. इसके अंदर microscopically you see myculic cells. ठीक है. So this I will teach you tomorrow. This is myculic cells, totally different. Very good. Someone said, ma'am, इसका जो nose होता है, that is also known as the woody nose, hebra nose. Yes, ये सब आज नहीं बात करते. We will talk about it tomorrow. But I hope you've understood that rhinoscleroma is a bacterial infection, whereas rhinosporidiosis, someone said, ma'am, how can it show spherules? Instead of spherules, they also write sporangia, by the way. Instead of spherules, they also write sporangia, which means the same. But then you're saying, ma'am, how can a parasite show spherules? Because that is what I'm saying now. Abhi tak to it was always considered as a fungus only. That is why they named it as spherules. Now they are saying that it has been reclassified into a parasite. But terminology to pehle ka chal raha hai na. ठीक है. Woody nose can, yes, rhinophyma can also show. So what we'll do, we will talk about rhinoscleroma tomorrow because abhi I don't have photos. I was not expecting this question. So wo wala photos I don't have where I want to show you the myculic cells and all. I've kept that as a question for tomorrow when we'll come to bacteriology. But tomorrow, now that I know this confusion is there, I will get this question again, and I hope you will not do the mistake of rhinosporidiosis being as a fungal / parasitic infection and rhinoscleroma being totally different. ठीक है? Okay. Can we move forward? Now going on to the next one. There's a 40-year-old पढ़ ही नहीं रहे. How many of you are just like me over here and you are not reading this and you are saying हमें answer पता है because we have seen something and we have marked it as chromoblastomycosis. Good, we are all on the same track. But exam में इतना confident मत हो ना, although you are right over here. Why did you call this chromoblastomycosis? Because we saw those brown color bodies called copper penny bodies. Examiner will either give you this photo, but what I feel is because you answered it in less than 5 seconds, examiner does not want that. He will not give you this photo. He will say histopathology shows, he will not even mention copper penny, not even give photo. He will say histopathology shows. What are the other names of copper penny bodies? They will be mentioned. Ma'am, the other names of copper penny bodies are MMS, medullary body, muriform body, sclerotic body. Examiner will say that the histopathology shows medullary, या muriform, या sclerotic. That is how you will pick up. Without the photo, it is chromoblastomycosis. Let me read further. There was a farmer who was struck with a wooden stick. So there is some kind of a trauma. A wooden stick could be on the hand, could be on the foot, but very commonly in these patients, we see that the foot is what is getting affected. So there is some kind of a trauma history that is going to be present in a farmer, and they will say that because usually farmers tend to walk barefoot, so while walking barefoot, the trauma ho gayi, and after that, their foot is going to show these lesions. If I zoom into it, I thought it was a cancer, because can you see that cauliflower-like lesion? Yes, our warts is what you would want to call this, but dermatology में warts को क्या बोलते हैं? Ma'am, warts are known as verruca. That is why this condition is also known as verrucous dermatitis, chromoblastomycosis. तो disease का नाम था, उसका clinical नाम क्या है ma'am? Because of warty verrucous dermatitis, but now disease done. Clinical name done. What is the microbiological organism? This was the previous year question in INI-CET and FMGE exam. So over here guys, there's no other way of learning it, and I'm sorry in advance. All the FFF sounding ones are going to come in it. For example, we have Fonsesia pedrosi, Fonsesia compacta, Exophylla, Phyllophora, Cladophylophora. I mean, there is no other way of learning it. All these Fonsacia, Phyla, Phylophora, Cladophylophora, all these FFF, oof, that is how you learn it. That is what comes down to chromoblastomycosis. Penny bodies are done. दूसरा नाम पूछेंगे MMS. Can you tell me the treatment? Because actually they don't ask you the treatment. Reason being that you will have to give multiple drugs. You will have to give amphotericin B, plus you will have to do a wide local excision. Wide local excision means you will have to do a surgery, plus some people because of cosmetic purposes might require a laser. That is also going to be one thing. So it is not just a single drug, it is going to be a multimodality approach that you will have to go ahead with, and then you will be able to come and treat the patient. Okay, coming to the next question over here. Which of the following organism exists as yeast form at 37 and hyphae at 25? Temperature change, to shape change. Which fungus organism is going to be like that? That yeast form at 37 and hyphae form is going to be at 25. Answer you're saying is what? Histoplasma. Okay. So what kind of a fungus is this that you're talking about? Ma'am, these are dimorphic. Dekho, do morphology. What is the 2-2 morphology that you're talking about? Yeast is at a higher temperature. Yeast kaisa dikhta hai? Yeast is going to be round like this. And what is mold? Mold is nothing but hyphae. Hyphae matlab it is going to look long like this. So can I learn that yeast it has a higher temperature, yeast heat 37, mold sounds like cold, so that is at a lower temperature 22, yeast is at 37 and mold is at 22, 25, yeast, heat, mold, cold. Perfect. So now tell me, someone said candida also. No, candida is not a dimorphic fungus. I think what you're getting confused is that candida can exist in yeast but not hyphae. I want to correct you. Candida mein true hyphae nahi hota. Candida mein the word pseudo hyphae will be written. If they would have mentioned yeast and pseudo hyphae, then I would have thought of candida. यहाँ पे hyphae लिखा था, so this is not the candida वाला thing. Now tell me, what are all the dimorphic fungus? Ma'am, you are saying that with the heat, the shape is going to change. So that means the mnemonic is also body heat probably changes shape. These are all the, see, mnemonics also we have made as per definition. Heat and cold hoga, तो shape change hoga. Body heat probably changes shape. Starting with, first you tell me and then I'll ask you one twisted question of PYQs. Number 1, B से क्या है? Blastomyces. अभी सबका photo. Now 10 minutes are the most important 10 minutes of fungal chapter. So blastomyces. H for histoplasmosis. P for Penicillium marnefi. P for paracoccidioidomycosis. I cut out the para. I'm left with what? C for coccidioidomycosis and S for what? Sporotrichosis. Now tell me, which out of these dimorphic fungus is also the fungus that you saw in subcutaneous layer? Subcutaneous का mnemonic था MRCS. If you remember, subcutaneous was MRCS. Do you remember Sporothrix, which you saw in subcutaneous, is also present in the dimorphic fungus? So this is going to be subcutaneous also, and this is going to be dimorphic also. Very, very good, and that's the PYQ that you have been asked. अभी के लिए out of all these, ये सब options आपके पास हैं. I want you to answer one question over here. Anyway, यहाँ पर तो histoplasma was the answer which you selected. Let's start with this question. I've got you from version X. Little lengthy. I hope you can read because that's the maximum font size I could get to. 45-year-old male patient presents with cough and fever for the past month. History of working at a construction site exposed to soil and bird droppings. Bronchoalveolar lavage showed microbial elements. Urine antigen is positive for the causative organism and histopathology is shown below. Which of the following structures is most likely observed? Okay, and ma'am, maybe हमें ना इतना कुछ नहीं समझ आया, but one buzzword that we picked up, fungus works on buzzwords. Soil and bird droppings is what we could pick up, and then we saw one big cell. We could see this big cell always ready to eat everything. The foodie is the macrophage, and macrophage has picked up these small. In the macrophage, हमेशा histoplasma decides I will go and sit. Yes, and bird and pigeon droppings, bird bat droppings. Yes, this is going to be histoplasma. Histoplasma means you're talking about a fungus. Fungus में क्या होता है? पेप्टिडोग्लाइकन तो मैम वो ग्राम पॉजिटिव बैक्टीरिया में था. लाइपोपॉलीसैकेराइड ग्राम नेगेटिव बैक्टीरिया में था. टेकोइक एसिड वाज पीपीपीटी ग्राम पॉजिटिव. Fungus is going to have chitin and beta 1,3-D glucan. Do you remember? And that is how they will club your systemic mycology with your general mycology. Between the same chapters, they will do integration. Now coming back to how did I pick it up? So what I'm doing, I have 2 flashcards, and in the last day of your revision, these 2 flashcards of the entire fungus chapter, so you have to revise. All the dimorphic fungus we have written. Your study is, you know the name of the disease, उसके एक्स्ट्रा नाम एंड उसका फोटो. जल्दी जल्दी, लेट्स कीप अ टारगेट ऑफ 5 minutes and finish off dimorphic fungus. When I talk about B for blastomycosis, over here what is the other name? Ma'am, it is known as North American blastomycosis, also known as Gilchrist disease or Chicago disease. And B looks like 8, so it is going to show me the figure of 8 appearance. Why is this looking black in color? Because what stain must I have used? The silver wala stain, that is Gomori methenamine silver stain. B for blastomyces, B for 8 jaisa, so figure of 8 appearance. Let's move on to, see, we are going in the same mnemonic. Body heat probably changes shape. So body is done, sorry. Body is done. Now we come to heat. H for what? Histoplasma. Do you remember? Hi, darling, see you in the cave. Who can forget that? Hi, darling, see you in the cave. Why did we learn it? Because hi will tell you for histoplasma. Hi will also tell you for hi, Ohio disease. They will always say patient is from Ohio in United States of America. Next, hi darling, it is also known as darling disease. See you in the cave, it is also known as cave disease. So hi darling, see you in the cave is how you are going to learn this. Now let's move forward. That you might feel that, oh, this is sounding like so romantic, but what romance in a cave? Nothing. You will come across bats and birds. So what will the patient be exposed to? That is going to be bat and bird droppings, and that is the question that you are going to get. No romance. So as the name suggests, it is going to be bat and bird droppings. See, wo question mein tha. Now once the histoplasma will go inside the body, I see two things. Ma'am, you had told us one very big cell which is known as the macrophage will go and eat it up. So ye chota chota histoplasma is eaten. Can I say this is the fungus that sits inside macrophages? Yes. Macrophage itna easy naam likhenge wo log? Bilkul bhi nahi. They will write the tougher name. What is the toughest name for macrophage that you guys know? That is reticuloendothelial cell. Imagine, itne simple se macrophage ko they call it reticuloendothelial cell. That is why they say histoplasmosis is also known as reticuloendotheliosis, which means that acha, oh, this is going and sitting inside the macrophages. It's the same thing, yeah, reticuloendotheliosis. Now when I look at it under blue color wala LPCB, this is the question that came in FMG exam also. It shows you what? It shows you tuberculate macroconidia, मतलब कुछ round होगा and उसके ऊपर छोटे छोटे projections like this. This is tuberculate macroconidia that you are going to see in cases of histoplasmosis. Khatam. Now body heat probably is what? Penicillium marnefi. Now instead of Penicillium marnefi they write Talaromyces marnefi, same cheese. As long as marnefi is written, it is okay. And marnefi reminds me of Marna. Whom are you wanting to hit? I mean, you're so bad. You're wanting to hit a rat. If a rat enters your house, you will run after the rat. How will you run after the rat, ma'am? I will take the first thing that comes in my hand is a jhadu, a broom. I will take a broom and I will run after the rat and I will hit the rat so much till the time the rat becomes red. So it is learnt as mar ke lal karna. Why did I tell you this story suddenly in the middle? Because if they ask you what is the reservoir, it is a bamboo rat. What color pigment is produced? It is a red color pigment. And what does LPCB show you? It shows you as if the conidia, these chota conidia are arranged in the form of a broom. So mar ke matlab marne fi, kisko? Rat ko. So bamboo rat is the reservoir. Lal karna is going to be the red pigment, and kisse? Jhadu se. So that broom is going to be the broom appearance for Penicillium marnefi. I don't think you're forgetting this story. Body heat probably. P say one more thing, paracoccidioidomycosis, which is known as South American blastomycosis. I hope you remember only blastomyces will be written, that is North American. Paracoccidio written, that is South American. And now tell me, paracoccidioidomycosis is going to show you number 1, pilot wheel appearance. It is also going to show you the Mickey Mouse appearance. It is also going to show you mariner wheel appearance. Same cheese bar likha. Matlab paracoccidio number 1 shows you Mickey Mouse. Mickey Mouse to dikh raha hai, ma'am. It shows those projections which are Mickey Mouse. Then it shows you this wheel. This wheel looks like the wheel of a pilot or a mariner. So pilot wheel or mariner wheel appearance is paracoccidioidomycosis. Coming to the next, para word hata do. Coccidioidomycosis. Please note, in coccidioidomycosis, I will write coccidioidomycosis. It is also known as desert rheumatism, desert valley rheumatism. Desert to samajh aa gaya. History will be of a patient living in United States of America in the Arizona desert. Arizona desert, California valley, कोई valley होगा, कोई desert होगा, सब ज़्यादातर USA का patient होता है because dimorphic fungus is very commonly seen there. So desert का history, and rheumatism means that joint involvement will be there. So desert rheumatism I have understood. Now they will show me these dabbas. These dabbas are known as barrel shaped arthrospores. These dabbas are referred to as the barrel shaped arthrospores, which is a very important question that they get. Now tell me, apart from barrel shaped arthrospores, arthrospores. For INI-CET students, can you also see these cup-shaped spherules in coccidioidomycosis? Yes. So instead of learning it as coccidioidomycosis, I can learn it as cup-shaped spherules that are going to be present. Repeating once again, barrel-shaped arthrospores with cup-shaped spherules are seen in desert rheumatism. Coming to the last one, body heat probably changes shapes, sporothrix. I always put a smile in front of sporothrix. Sporothrix is that dream life that all of you are studying for. I don't know whether we as doctors will achieve that life, but that is the dream life that everyone has, irrespective of whichever, a kind of dream, ultimate goal you may have professionally, but your dream life will always be this. And your dream life will be that you would always want that you are roaming around. Let me just give you a snapshot of your dream life, especially the ones that is shown in Hollywood movies. That, ma'am, I am not surrounded by an AQI of 500. I am roaming around in the open fields and gardens. I have so much of time in life. I am able to see the stars at the night. I'm going to go camping. I'm going to see the starry sky. Then, of course, as per Hollywood movies, that we don't promote it, but as per Hollywood movies, the good life is also that you're sitting under the stars and you have a cigar in your hand. So imagine that kind of life that you are wanting to live, a fungus is living. And that fungus is Sporothrix shenkai. Whom are you jealous of? Imagine that day has come when you're jealous of a fungus, and that is Sporothrix shenkai. So what is it? Rose. It's working in the rose fields, rose gardener's disease. They will say there's a rose gardener with a thorn prick. You think of Sporothrix shenkai. So now if the thorn prick has happened on the fingers, did you notice that from the finger, the lesion has started spreading along the entire arm? It might also go along the leg. So wherever it's spreading, ultimately it is spreading how? It is spreading via the lymphatic channels. It is a lymphocutaneous spread. Now you want to look at it under the microscope. Under the microscope, it looks like a rose being made over here. So it is going to show you rosettes of conidia. Another rose garden mein hoga, rosettes dikhega. Next, it shows you a round structure in the center, which I'm drawing again. That round structure is known as a cigar. ऐसा round वाला cigar कौन सा होता है, ma'am? They basically want to show you the pencil like this, but they have turned it and shown it to you like this. So that's why it's looking like a dot, and that is a cigar, and it is surrounded by this starry thing. So that is known as asteroid body. Center wala is cigar body, and surrounding it is asteroid body. So you will say, what are all these? The center is the cigar, which is the fungus. The center wala cigar is the yeast. It's the fungus, Sporothrix shenkai. What is surrounding it? This is a very important phenomena that you are asked in dermatology. Splendor Hopley phenomena. Very, very good. Splendor Hopley phenomena. क्या होता है? Ma'am, it is nothing but antigen-antibody reaction. No one will ask you क्या होता है, but I am telling you because you can't mug up everything. It is antigen-antibody reaction which has occurred over here. So now the good life. Rose garden, rosettes of conidia, cigar in the hand, stars in the sky. That is Sporothrix shenkai. So if I ask you a question, let's see, without options, are you able to solve? I don't think you're reading this. I don't think you're reading this. I think your answer is in front of you. As soon as you saw the figure of 8 appearance, your answer is, ma'am, this is blastomyces. कोई doubt ही नहीं है. But yeah, blastomyces means it is causing all of lung wala involvement. Plus you said blastomycosis is known as North American blastomycosis, that is fitting in. But apart from that, it is also showing you skin lesions. And yes, these infections can show you skin involvement also. Derma hota hai. Now coming to one more question. What do you want to give the answer to this? इसमें I have given you options, but I don't think you're reading. See, there's a gardener. Gardener आते ही साथ में मैम rose gardener and rosettes are seen over here. Answer is clear. It is sporotrichosis. What did I tell you? That sporotrichosis spreads via what? Via the lymphatic channel. That is also mentioned over here. So nothing tough that we had. Okay, one more thing that I want to ask you. A quick question is what you were mentioning me कि asteroid body और कहाँ देखने को मिलता है. तो asteroid is a star which is seen where? Sporothrix shenkai. It is also seen in sarcoidosis. And how do you differentiate? Like Dr. Pawan is asking. So very simple, Dr. Pawan. Sarcoidosis is a disease which has granulomas, non-cageating granulomas, which basically means it has giant cells. So if you will see a big giant cell and the star will be inside that giant cell. So this is going to be intracellular star inside a giant cell. Whereas Sporothrix is a rose gardener. Gardener will always roam outside. So this star will be lying around outside. It is going to be an extracellular star. Is that okay with everyone? So I can say star is seen in Sporothrix and sarcoid. Sarcoid mein andar, it's intracellular. Sporothrix is rose gardener. Rose gardener will be roaming around outside. Okay with everyone? Now I'm going on to the next question. Something on this is expected in the exam. 55-year-old man presents to the hospital with cough, trouble in breathing, chest tightness, fever. Known case of HIV, currently taking antiretroviral therapy. Complaining of neck pain and headache, and then in an HIV patient when neck pain or neck rigidity, something like this is mentioned, obviously my doubt towards meningitis always goes. There is also an X-ray done because he initially had all these cough wala symptoms. Cavitatory lesions are also seen. Grab staining shows yeast morphology. Yeast मतलब true yeast से हमेशा एक ही आता है, and that is very good. That is cryptococcal meningitis. Perfect. All of you are marking cryptococcal meningitis. Perfect, guys. Now, what are the questions on cryptococcal meningitis? Which cryptococcus are you talking about? Because there are two. One is known as Cryptococcus neoformans and one is known as Cryptococcus gattii. And this is the recent FMG exam question that came last year, जो January वाला मतलब अभी recent session हुआ था. Now they will ask you that okay, which out of these are going to be associated with pigeon droppings? So Cryptococcus neoformans is associated with pigeon droppings. In fact, that is the most common one also. That is the one which is most pathogenic. Whereas Cryptococcal gattii is going to be seen along Cryptococcal gattii tree. This is seen in the soil around the eucalyptus tree. This is seen in the soil around the eucalyptus tree. So remember, gattii is the soil around the eucalyptus tree, whereas neoformans is going to be around the pigeon droppings. Next, which of them are going to be seen in tropical and subtropical countries? Ma'am, gattii is going to be seen in the tropical and subtropical countries. That is one thing. यहाँ तक याद हो गया? Now I start with the next thing. Let's say cryptococcal neoformans is सबसे common. So they will ask you what all serotypes come under Cryptococcus neoformans. तो मैम Cryptococcus के serotypes होते कितने हैं? A, B, C, D, E, ऐसे बहुत सारे serotypes हैं. Usually they consider 4 like A, B, C, D. Out of them, please note the first and the last one usually come under Cryptococcus neoformans, whereas the center two ones gattii में आता है serotype B and C. Okay. Now coming back, what is this going to cause? You will start fill in the blanks for me. Ma'am, it is going to cause meningitis. Meningitis matlab I will take a CSF sample. CSF sample लिया तो कौन सा stain? This is what someone was asking me in the first 1 hour of the class. कि ma'am negative stain बता दोगे क्या होता है? Negative stain is like the negatives that we used to have in the old days in cameras. Like what happened in that is the background. See, when I say you are staining something, staining मतलब let's say staining gram positive bacteria, तो मैम gram positive bacteria got the purple color. Background को थोड़ी ना color दिया, you gave the color to the bacteria. Here you are doing opposite, here you are doing negative, here you are giving the color to the background with the hope that okay, the organism, because it has not got the color, then the organism will shine out. So can I say that over here the background is the one that is going to be stained, but then you are also saying that the organism over here will shine out because it is not stained, it is going to be unstained. So please remember, background is stained and organism is unstained. That is negative stain. Example, India ink and nigrosin. That is the CSF. Meningitis था CSF sample लिया ऐसा करा. But if you took a biopsy sample, for a biopsy sample, who does a brain biopsy for meningitis? No one. But they will ask you a question. Unless the patient has died, autopsy हुआ है तो then तो brain biopsy would be done. Can I say for a living, just a very brutal, crude way of learning, कि मैम living patient में when CSF is taken you do negative stain, मरा हुआ patient you do mucicarmin, because मरा हुआ patient is when I would do a brain biopsy, otherwise who will do a brain biopsy? So for मरा हुआ patient we will do mucicarmin. Very crude way of learning. Now let's come to the culture media. Because neoformans, ma'am, this was to do with pigeon droppings, birds. So the culture media is also known as bird seed niger, and niger word will tell me that it gives a black color, brown color, वो तो obvious. But bird seed niger will be for Cryptococcus because this is associated with the pigeon bird. Now, which enzyme does Cryptococcus make? Cryptococcus makes urea or urease. It is a urease positive organism. Enzyme. And this question I'm sure everyone has seen. Earlier it was said that the gold standard method is latex agglutination test, but now genetics is superseding everything. If they write LFA, lateral flow assay is written in the options, that is a better diagnostic modality than latex agglutination test. So if lateral flow assay is mentioned, Best answer. If not mentioned, latex agglutination test. Now coming to the treatment, ये तो PYQ है. Every word that I have written over here is a previous year question. Don't blame me later on कि मैम ये इतना ज़्यादा डिटेल में जाके पूछा था. Everything is written over here. You have to know every word. What is the treatment? It is liposomal amphotericin B plus flucytosine. Yes, that is liposomal amphotericin B plus flucytosine. यहाँ तक तो question आया, but this is the initial therapy that is given. In fact, when you go through medicine, they will say that in the later stages, fluconazole is also given. In the later maintenance stages, fluconazole is also going to be given. So liposomal amphotericin B plus flucytosine तक ही question आता है, but in the later stages followed by fluconazole. That is the complete treatment that I want to tell you. Finally, this is done. Let me ask you another question. 35-year-old immunocompromised male comes to the emergency department. Immunocompromised, probably HIV positive, headache, photophobia, altered mental status. They are thinking of meningitis because they have done a CSF analysis and they reveal an encapsulated yeast cell. The negative stain is performed. अच्छा मैम, now तो I know negative stain मतलब cryptococcus सोच रहे हैं. Which of the following best describes what is stained, what is not? Negative मतलब उल्टा मैम, negative background was stained. So can I say the yeast cells will appear colorless and the background will appear stained? This statement, see, think of your statement. What was colored? The background was cleared, the organism was not. This is the best answer that is going over here. Are we okay with this? Moving on to the next question. There is a 17-year-old girl who presented to the emergency room with toothache on the left side of the face for 7 days, with subsequent development of pain and swelling with periorbital edema. Known case of type 1 diabetes. The culture report confirmed the presence of aseptate hyphae. See, anyway in immunocompromised state like diabetes and with periorbital facial involvement, mucor is what I was anyway thinking of. Plus they said aseptate hyphae, तो मैंने भी सोचा mucor once again, which means that they will always ask me the difference between aspergillus and mucor. यही इनका घिसा पिटा question which they always come up. I will give you 2 minutes for aspergillus, 2 minutes for mucor. When I'm talking about mucor, tell me, let's start with aspergillus. That's an easier way of learning. Ma'am, aspergillus mein we always have acute angle branching. And what kind of branching are there? Septate. So when we will do the branching, it will always be 45 degree and the hyphae are going to have septa. That is aspergillus. So let's zoom into it. What is the branching angle? The branching angle is acute angle, and look at the septa that are coming in between. ये aspergillus है. Versus mucor is completely opposite. Mucor will be what? Branching. Ma'am, opposite. मतलब it is going to be 90 degree obtuse. Acute नहीं होगा. It is 90 degree or obtuse. And there are no septa. It is completely clean. Can I say mucor is going to be 90 degree and it is going to be aseptate? And that is what was asked in the question, aseptate. So mucor. So now coming back, if they ask you which of them out of the two is looking thicker, mota. So remember, mucor is mota. That's another way of learning it. Examiner won't write mota. So examiner will write which of them is a thick ribbon-like fungus. So mucor is mota. It is a thick ribbon-like fungus. Very good. This is done. अब दोनों का एक-एक word बता दो. Because this question was to do with mucor, this is that famous black fungus that we had. Is it a real name, a true name, black fungus? Yeah, I should write it and I should cut it off, because black fungus is what the new social media and news channels called it. Patient was coming to you with black color necrotic lesion, इन्होंने कहा black fungus. It is not a black fungus. Next, I have told you microscopically it is mota, it is 90 angle, it is aseptate. So that is also what we settled. Now we come to what does it grow on SDA. Ma'am, on SDA, it tends to be known as lid lifters. Lid lifters means that if, for example, this is my culture plate, and on the culture plate, obviously I will put the mucor and I will put a lid on top of it. After few days, I will notice that ये जो lid है ये उठ गया. Someone has lifted the lid because it becomes like cotton. Like cotton, it is growing inside, and this cotton is basically going to lift the lid. That is why they are known as lid lifters. That is what is important. So this is for mucor, is what they will ask you. What will they ask me for aspergillus, ma'am? They will ask you the three variations for aspergillus. Firstly, how does aspergillus look under the microscope? Acute angle branching, septate, पढ़ लिया. What are the three types of aspergillus? Fumigatus, flavus and niger. I don't think तो पहला photos में क्या दिखाया? Culture media होगा, मतलब SDA culture media. Second photos में क्या दिखाया है, ma'am? The lactophenol cotton blue. Now when I notice SDA, something comes black. When I notice LPCB, something comes black. So that of course is going to be aspergillus niger. I don't think we have any doubt in it. We will just have to go for fumigatus versus niger. Fumigatus में क्या color आया? Smoky green color, because fumes and smoke go together. So fumigatus will be smoky green. Flavus sounds like a flower. So think of pretty colors, flower wala colors. Flowers wala colors is going to be yellow green wala color. In fact, it looks like a kiwi fruit also to most of us. But coming back, fumes and smoke ek taraf, flavus flower is yellow green. When how will they look under lactophenol cotton blue? Ma'am, the flower is going to look like a complete flower. Means the conidia are going to be all around. Ye conidia like rose petals, they are all around. Whereas in fumigatus, the conidia are only on the upper part. See, fumes will always rise up. So the conidia are only on the upper part, and that means that is fumigatus. So remember, fumes smoky green, conidia like fumes will only rise up. Flavus is flower, so yellow green, and like a flower, the conidia are all around. Niger is everything black black. A clinical question, patho ka clinical question I'm asking you here. What do you mean by aflatoxin? This you will get in medicine also, and I'm teaching you once for all three subjects. What is aflatoxin? Ma'am, A tells me aspergillus. Going by the three options, I think this is aspergillus flavus, and this is the toxin for aspergillus flavus. Okay, how will I end up getting this inside my body? This is said to be a groundnut contaminant. If you end up eating any walnuts, groundnuts, peanuts, anything that is contaminated with fungus, then that is this fungus. When it enters into our body, all of you are saying सबने अलग अलग मुझे answers दे दिए. I'll put it under one roof. This aflatoxin is going to go and cause a mutation in codon पूरा एक एक अल्फाबेट एज इट इज क्वेश्चन में आएगा. कोडॉन 249 of which gene? Of the P53 gene, policeman, and thereby it causes what? I think now everyone knows, it causes hepatocellular carcinoma. तो hepatocellular कौन से याद रहता है? P53 जीन भी याद रहता है. भूलने वाली चीज इज कोडॉन 249 because options are 129, 249, 279, 289 and then you are gone for a toss. हम तो याद करके गए थे, एग्जामिनर ने options इतना close दे दिया that we forgot. So when you write P53, you know how to learn this, na? याद आ रहा है from rapid revision videos. You know the fill in the blanks 2, 3, 4, 5 बनाना है. So 2 is missing, 3 is written, 4 is missing, 5 is written. P का हम 9 करेंगे, that's the only number I can think of. So 249 codon I have got. So बीच के numbers, yes, everyone remembers. So ma'am, we are going to do 249, not 129, 127 in the exam. Okay, all aspergillus questions are done. Coming to the next one. What is the organism that can be identified with the man who received a kidney transplant and is on immunosuppressant, because every transplant patient is, shows budding cells with pseudohyphae? This is what you guys were asking me in the beginning. Pseudohyphae दिखेगा तो क्या सोचने वाले हो मैम? pseudohyphae दिखेगा तो we will think of candida. So that means you will now tell me all the candida questions in one place. This is your flashcard. Mycology works on flashcards. फोटो कर लिया तो mycology हो गया. So when I talk about candida, number one you said छोटा छोटा जो दिखेगा, that is going to be the budding form, and then the long ones that you notice, those are what? Those are known as the pseudohyphae. So budding pseudohyphae done. Coming to, someone's asking me again and again about aspergillus. Is aspergillus angioinvasive? Yeah, and that is why we call it like again and again I've been telling you, invasive aspergillosis. So yes, it goes into the blood vessel. It is angioinvasive. Coming back, now coming to SDA. SDA पे क्या? Candida में ma'am, the colonies that you see, they are creamy, pasty, yeasty colonies. Very good. Creamy, pasty, yeasty colonies. अच्छा, candida के different members, if I have to differentiate, which agar will I use? I will be going ahead with the chrome agar. So this is also done. You can see chrome color, अलग-अलग colors नहीं याद करने. Coming to the next one, what is this photo? Ma'am, this photo I'm seeing spores, and those spores are double walled. ध्यान से देखो, दो walls हैं उनके अंदर. The spores are double walled. So what are these known as? These are known as chlamydospores. When will they make chlamydospores? On a special type of an agar known as corn meal agar. Okay, one photo that I have not shown, which INI-CET students will tell me, pap smear पे कौन सा वाला effect दिखता है candida का? Gynae question, that is known as shish kebab appearance. Did you notice how for candida we all became Shah Rukh Khan without knowing it? Did we become creamy pasty? So remember, candida shows you creamy pasty yeasty colonies, shows you chrome agar, shows you chlamydospores which are present on corn meal agar. Pap smear pe shows you kebab effect. All K is coming together. The only extra thing that you have to know is, very good, the Reynolds-Broad phenomena. The Reynolds-Broad phenomena, also known as, बहुत टफ नाम था मैम, रेनॉल्ड ब्रॉड फिनोमिना को जर्म ट्यूब टेस्ट कर देते हैं कि कैंडिडा में से द ट्यूब विल कम आउट, दैट इज नोन एज द जर्म ट्यूब टेस्ट. नाउ माय क्वेश्चन इज दैट ये दोनों जो चीज़ें आपने लिखा, the germ tube test or Reynolds Broad phenomena positive or the chlamydospore, it is shown by whom all? It is shown by Candida albicans, and then it is also shown by uska ek double brother, double cousin brother, dubliniensis. Candida dubliniensis also shows. So dubliniensis says whatever you do, I will do double double. If you do germ tube and chlamydospore, I will also do germ tube and chlamydospore. So if I ask you a question, which of the following tests is used to identify, or this test is used to identify what? So you can see na, एक fungus है जिसमें से germ tube is coming out. The best answer that I have over here is Candida albicans. If dubliniensis would have been mentioned, then I would have selected dubliniensis also. So now the question is that which of the following is false regarding Candida dubliniensis? Can it be cultured on corn meal agar to produce chlamydospores? Germ tube test is positive. Can it grow in hypertonic saline? And it cannot grow at temperature above 45 degree. Which answer are you selecting? I'm getting A and C as answers. See, I've asked you the false statement. So the false statement is this. I want to tell you that when you are differentiating between Candida albicans and Candida dubliniensis, you need to be very certain that number one, दोनों में germ tube test positive आने वाला है. Both positive. Then ma'am, दोनों में chlamydospores are going to come positive. That is also okay. So then we say to differentiate who's the OG. The OG elder brother is Candida albicans. So all the tough situations in life, the elder brother is going to step up. If I increase the temperature above 45 degree, who will grow? Candida albicans will go. If I put it in hypertonic saline, who will grow? Candida albicans will grow. Dubliniensis younger brother says no responsibility. I will not grow above 45. I will not grow in tough situations like hypertonic saline. So can it grow in hypertonic saline? It cannot grow in hypertonic saline. That is the false statement. Okay with everyone? Candida ki story bhi khatam. The last fungus that we have is a pharma question which you all know. What is the drug of choice for Pneumocystis carinii? They will not write it. They will give you the photo. So you need to know this is Pneumocystis carinii, and you all know the answer is going to be cotrimoxazole. तो अभी हम क्या रिवाइज करने वाले हैं before we end mycology? And many of you were asking me is parasitology also today? Yeah, session is till 3 o'clock, which means जितना पूरा तो नहीं होगा, but जितना I can do, because if tomorrow I make you do parasito plus bacterio plus viro, I don't know about you guys, but I will die in that process. So I can't leave so much for tomorrow. Parasito also at least over a half we have to finish today. Anyway, coming to Pneumocystis carinii. Why not pentamidine? Dr. Rafa is asking. हाँ दे सकते हैं pentamidine, but that is given in severe cases ना. So listen to me. When I talk about Pneumocystis carinii, you write PCP 4 times and you are done with 4 subjects. PCP means it is Pneumocystis carinii pneumonia. This means it is a kind of a pneumonia which is usually seen in HIV positive individuals when the CD4 count goes less than 200. That is one thing. Next, it is also known as PCP, means plasma cell pneumonia. When the pathologist will look at it under the microscope, what will pathologist look at? Pneumonia का patient है. Pneumonia का patient मतलब मैम या तो sputum आएगा, but इनमें sputum does not come. These patients have what? These patients have dry cough. Anyone who has a dry cough, I would want to take a bronchoalveolar lavage in that situation, and bronchoalveolar lavage is going to be showing me under the microscope plasma cells. Patho हो गया. Now let's come to microscope of microbiology. It shows you the crushed ping pong ball appearance. If you look at this photo, guys, why is this looking black? कौन सा वाला stain होगा? Is this the gram stain? Is this the pass stain? Is this the GMS stain? Black color मतलब GMS stain, Gomori methenamine silver stain. So I can see the black color. I can see these round balls, but at some places I can see the ball has been crushed. So this is known as the crushed ping pong ball appearance. Finally, coming to pharmacology, how do you do the treatment? The drug of choice for pneumocystis, drug of choice being asked is cotrimoxazole. Severe cases mein I could give pentamidine. That is how we learn it. So we are settled now, done with this, which means thank you for all the patience. Mycology pe maine cross lagaya, khatam kiya. Not that you end the session. You are still going to sit with me for 35 minutes more, whatever. I promise after 3 o'clock I will not extend, but 3 o'clock tak jitna parasitology we can do, let us wrap it up. Are you up for it? Yes guys, good to go. Can we start? No break, बिल्कुल भी नहीं, not at all. No break. Every time. See, this happens in every session, every LRR. General mycology and half of parasito happens on one day. Then helminthology wala I teach you on the next day, because those adult worms you stop understanding by 3 o'clock. So adult worms, and then bacteria and virus ke sare flashcards I will teach you tomorrow. Okay. Of course, I was just asking for formality. Even if you would have said nahi baithenge ma'am, I would have said baithna padega. Let's start with the first question. A 34-year-old patient presents to to the clinic with complaints of bloody diarrhea, fever and abdominal pain after having a recent traveler, maybe traveler's diarrhea हो रहा है, travel history है, माइक्रोस्कोपिक एग्जामिनेशन बता दिया, थैंक गॉड. एंटमीबा हिस्टोलिटिका पूछा क्या है, ट्रीटमेंट रिजीम. ट्रीटमेंट रिजीम में मैम मेट्रोनिडाजोल आंसर करने का मन करेगा, पर मेट्रोनिडाजोल के आगे पढ़ना जरूर, बिकॉज समवेयर मेट्रोनिडाजोल फॉलोड बाय पैरोमोमाइसिन विल बी देयर. बाय मेट्रोनिडाजोल यू आर कंट्रोलिंग 10 days मेट्रोनिडाजोल, डायरिया कंट्रोल हो जाएगा. बट व्हाट अबाउट द पैरासाइट हु इज हाइडिंग इन द इंटेस्टाइन एंड सिटिंग and who will come back later with another attack of diarrhea? So paromomycin is being given because this is a luminal amebicide. Don't miss this. It has to be metronidazole plus paromomycin. Don't get confused with only metronidazole. So now when I'm saying entamoeba histolytica, these two photos to you all know na. Entamoeba histolytica, what will it cause in the intestine, the sabka favorite flask shaped ulcer, and what will it cause in the liver? Ma'am, liver is going to melt like brown color pus, that is known as the anchovy sauce appearance. No one can get these two questions wrong. But if they ask you inside the flask, what is seen over here? ट्रोफोसॉइड दिखेगा या सिस्ट दिखेगा? इनसाइड द एंकोवी सॉस अपीयरेंस माइक्रोस्कोपिकली क्या दिखेगा? सो प्लीज नोट, फॉर द डायग्नोसिस इन द पस वी सी ट्रोफोसॉइड एंड दैट इज अ प्रीवियस ईयर क्वेश्चन. सो नाउ व्हाट, to save our time, wherever I write T, that is trophozoite, and wherever I write C, that is cyst, because if I keep writing these 100 times, then our session will go until 5 o'clock. So T for trophozoite, C for cyst. ठीक है, अब हम एंटामीबा हिस्टोलिटिका का trophozoite versus cyst देखते हैं. When I look at these, I notice the trophozoite has how many nucleus? Of course, it has one nucleus, but that nucleus I can visualize has a dot in the center and some dots on the side. Has it started looking like a cartwheel appearance? Has it started looking like a cartwheel nucleus? They rarely ask you this question. Apart from the nucleus, I can see red blood cells. Okay, it has eaten up red blood cells. The only parasite to eat up red blood cell is Entamoeba histolytica. बस उसके नाम में था ना, in case you didn't notice, histo means tissue, litica means breakage. It is breaking the tissue, eating the blood. Breaking the tissue, tissue break होगा तो blood loss होगा ना, it eats that blood. That's its like guilty pleasure that it has. So it eats the blood. Wherever it breaks, it eats the blood. Only parasite to show erythrophagocytosis. Now this is the trophozoite. Let me come to the cyst form. The cyst form has how many nuclei? 1, 2, 3, 4. It can have between 1 to 4. That doesn't mean हमेशा 4 होंगे मैम. Sometimes there'll be 1, sometimes it is divided into 2, sometimes it will have 4. So between 1 to 4 nuclei will be there, and there is a very very thick body which is known as the chromatoid body. For INI-CET पहले तो for all exam questions, chromatoid body किस चीज से बना होता है मैम? It is made up of ribonucleoprotein. It's a ribonuclear, nucleic acid वाला protein. So ribonucleoprotein. One question for INI-CET students: what is the stain for chromatoid body? And that is going to be iron hematoxylin. It is going to be iron hematoxylin. That's only for INI-CET. Okay, now moving, although I was telling for patho also, whenever I say only for INI-CET, I expect students would sleep off. Somehow your mind becomes more active, and even FMG students end up learning it. This is how just the human brain works. For some odd reason, what we are told not to study is what we end up studying. So coming back, flask shaped ulcer, anchovy sauce appearance, trophozoite में एक nucleus, cyst में 4 nuclei. Nucleus याद करते जाओ. After 10 minutes, you will make a table where you will write all the number of the nuclei of all the parasites. So Entamoeba histolytica, number 1. Trophozoite, how many nucleus? 1. Cyst has how many nucleus? 4. I want you to compare this with Entamoeba coli, just the number of the nuclei. In Entamoeba coli, the trophozoite again has 1 nucleus. So Entamoeba histolytica का and Entamoeba coli का मैम trophozoite is going to be 1, 1. Simple, 1, 1 nucleus. The cyst is where it's different. In Entamoeba histolytica, it is 1 to 4. In Entamoeba coli, it is going to be 1 to 8 nuclei. Nuclei याद करते जाओ. Coming to the next question. A 32-year-old male patient presents with complaints of loose stool, fever and abdominal pain. Every parasitology question has to come to you with loose stools. Diarrhea तो सभी को हो रहा होगा. Gone for a trip to another city. Patient is otherwise stable and general physical examination is also normal. The stool examination reveals a ciliated parasite. There is only one ciliated, cilia, coli के अंदर cilia, the only ciliated parasite. So Balantidium coli is the most likely organism, is what you are trying to tell me, which appears true. So I will come back to this question, but every flashcard detail about Balantidium coli in one page. पूरा 10 minutes का rapid revision in 1 minute you have to study. When you write Balantidium Belantidium coli. Ma'am, the word coli will tell you about celi. This is the only ciliated organism, and that is what I taught you in the first 1 hour also. Okay, motility hota hai. Now I can tell you which kind of a motility. It is a rotatory motility. So it can show you rotatory motility because of cilia. Now B for Belantidium, B for big, bada. It is going to be the largest protozoa of our intestine. Which intestine, ma'am? Bada intestine means the large intestine. How many nuclei does it have? Binucleated. So whether it is a trophozoite, 2 nuclei. Whether it is a cyst, 2 nuclei. Abhi tak to you were saying that, ma'am, trophozoite has 1, so cyst has 4. Trophozoite has 1, so cyst has 8. Cyst always had more number of nuclei. This is the first parasite where you're teaching us that there is no change in the number of nuclei in cyst and trophozoite. Both of them have 2 equal to 2, which is that parasite. It is going to be Belantidium coli. Giardia का पूछ रहे हो? No, why should I tell you? I will teach you Giardia afterwards. You will answer that nuclei wala question. It is not my test, it is your test, but we will come to Giardia in a while. When I talk about binucleation, okay ma'am, one big nucleus, one small nucleus. The big one is macro, the small one is micro. The big one, the macro one, looks like a kidney. So if you look carefully over here, you will notice there is one big nucleus which is looking like a bean, and there is one small nucleus which is looking like a dot. So now if you put everything in perspective, B for bada wala protozoa, B for bada wala intestine, B for binucleation, out of which the big nucleus is going to be bean shape. Now this is going in the intestine. So what is the clinical feature in the patient? Ma'am, patient will have diarrhea, and for diarrhea, of course, intestine mein to diarrhea hoga, and for diarrhea the drug of choice is doxycycline. So now let me come back. Ye sab karne ke liye there are 2 forms, trophozoite and cyst. Whenever I ask you who causes infection, whether it is for Entamoeba histolytica, whether it is for Giardia lamblia, whether it is for Belantidium coli, infection hamesha kaun sa infective form is usually the cyst. One place trophozoite will be the infective form, T for T, that is Trichomonas vaginalis. Trophozoite for Trichomonas vaginalis, baki sab tab tak aap cyst karo. So coming back, cyst is not the infective stage of the organism. Galat. Cyst is the infective stage. Is it the smallest protozoa? Galat. Big, bada, biggest, largest. Does not show motility? Galat. It is ciliated. It is going to show rotatory motility. So this is going to be the only correct statement that we had over here for ciliated organism. Moving on to the next question. There is a 28-year-old female who presents to the physician with eye pain. We moved away from diarrhea. She has a history of contact lens usage. Whenever they say contact lenses and eyes, एक organism comes to my mind. Actually, 2 come to my mind. One is parasite पूछा तो acanthamoeba with contact lenses, and if bacteria is asked, तो pseudomonas definitely comes to our mind. After a complete evaluation, there is severe retinitis also, and then the culture of the water was done. The possible pathogen that would be isolated is going to be acanthamoeba. तो मैम यहाँ पे बैक्टीरिया तो बोला ही नहीं, pseudomonas तो है ही नहीं, तो acanthamoeba. So now what is acanthamoeba? It's an amoeba, obviously, but it is a free living amoeba. It is not present in the food and etc. It is present openly in the environment. It is not coming from any animal or a life cycle. It's openly in the environment. So there are 2 free living amoeba they will ask you. One is Naegleria fowleri and the other is acanthamoeba. Naegleria fowleri ka story to everyone remembers. Nala, nahana, naak, neural, which means Naegleria happens because of taking bath in dirty water. So nale mein nahana. Examiner will never write someone went into a gutter to take bath. Examiner will somehow, so bias, like patient is in South India, so matlab he's forever bathing in ponds and he's having rhinosporidiosis. Patient is a truck driver, means he's having unsafe sexual practices, syphilis kara dena. Patient is going to his village for a 5-day vacation, तो examiner assumes village में bathroom नहीं है, ऐसा उसका assumption है that village is only going to have dirty water and the patient will take bath in dirty water only. So visit to the village, assume he's taking bath in dirty water. So नाले में नहाना, okay. After that naak, where will the water go? The water will go into the nose. From the nose, what is the plate over here, guys? कौन सा plate होता है? That is cribriform plate. Via the cribriform plate it will go into the brain. So neural. So remember nala, nahana, naak, neural. If that story comes, it is Naegleria fowleri. So this is because of swimming and the spread is going to be neural. The disease called is PAM, that is primary amoebic meningoencephalitis. Meningoencephalitis, that is what it causes. Now coming to acanthamoeba. Ma'am, acanthamoeba is opposite. It is not associated with swimming. It is seen in HIV positive individuals. HIV goes in the blood. This parasite also goes in the blood. This is not the nala, nahana, neural story. This is going into the blood. blood and this causes granulomatous amoebic encephalitis. One patho question, jaldi jaldi. Granuloma is acute inflammation, chronic inflammation. Give me a quick answer, give me a correct answer. Acute inflammation, chronic inflammation. Ma'am, granuloma is chronic? Yes, I hope. Chronic inflammation. So they will ask you which of them is going to have a chronic course. So automatically acanthamoeba, which of them is going to be acute and fatal? Acute and fatal very, very quickly. They will say patient has ended up dying in 5 days of having meningitis. That is Naegleria fowleri, whereas granuloma wala is chronic. Acha, if it is not causing granuloma, then it is gone and caused the eye wala problem. Contact lens ka users may cornea ulcer problem, which was asked over here. Now I want to see some photos. They don't give you photo of Naegleria fowleri, they give you photo of acanthamoeba. What is the meaning of the word acantho? Before I start, where in pathology did you study acanthocyte? Site matlab cell, acantho. Ma'am, there were RBCs with spike. A for acanthocyte, A for A beta lipoproteinemia. We have studied them, and then I gave you homework also on day 1. Then day 2 you told me the mutation of A beta lipoproteinemia, which was MTTP gene mutation. ये सब पढ़ चुके हैं, which tells me wherever the word acantho comes, the word spike comes, and that is why when I see the trophozoite, I can see spikes coming out of it, which is so nicely seen. That is the trophozoite of acanthamoeba versus the cyst of acanthamoeba that you have over here. Ma'am, the cyst is just like any other cyst, but it's not round the way you've drawn it. The cyst is having a wrinkled wall. Can you all see? As a zigzag wala wall hai. So please note that over here spikes are going to be there in trophozoite, and there is a wrinkled cyst wall that I will see, and now I will know they're talking about acanthamoeba. Why did I teach you? First, coming back, yes, there is someone had asked me with eye pain, retinitis is possible. Retinitis, and they are also saying that after a complete evaluation, a diagnosis was bit apart. Okay, that's your question. Okay, Dr. Harsh, corneal ulcer will be the primary thing that you will always think of because of contact lenses, keratitis, corneal ulcers is what we suspect. But over here, even if it has resulted in retinitis, out of all of these, the possible pathogen that can come from free living, means tap water, and via contact lenses will anyway be acanthamoeba. So it's not that after corneal ulcers it cannot cause retinitis. Maybe it's a second down in the list of clinical features. Now coming back, this was the question that came in INI-CET. A person from a rural area experiencing CNS symptoms and vision loss for the past 12 days. Matlab probably granulomatous amoebic encephalitis and there is some keratitis occurring over here. Electron microscopy पे मैम मुझे ऐसा ऐसा spike दिख गया, spike दिखा तो I will think of acanthamoeba. See, initially when I saw rural area मुझे लगा नाला नहाना kind of a story, but then when they came up with the photo of the spike, I'm like no, spike will be there in acanthamoeba, not in Naegleria. Acha, what is this third one mentioned? What is B. mandrillaris or Balamutia mandrillaris? So this is for INI-CET students. Again, do you know Balamutia mandrillaris is acanthamoeba ka cousin brother? Which means that if acanthamoeba causes granulomas, then Balamutia also causes granulomas. Difference hai that the trophozoite is different. Na acanthoma to ma'am it will show you spiky. That is the photo that you see over here. Whereas B. mandrillaris shows you a trophozoite which is going to be a, yes, a, what is it? Fish shaped. Now try to visualize this fish. This is the fish shaped trophozoite that we have over here. This is the fish shaped trophozoite that we have. Otherwise, minor other differences are there, but this is the predominant one. Both of them can cause granulomatous amoebic encephalitis, and this is what it is going to result in. Someone said that ma'am 12 days over here is also acute. Agreed with you. This is why this was INI-CET exam question and created so much of confusion. People said rural area का हिस्ट्री था, 12 डेज का हिस्ट्री था, मैम अक्यूट सा लग रहा था, जल्दी जल्दी हो रहा था सब कुछ, मे बी नाइग्लेरिया. बट देन व्हेन यू हैव एन इलेक्ट्रॉन, मतलब नॉट इवन लाइट माइक्रोस्कोप, फॉरगेट दैट, इलेक्ट्रॉन माइक्रोस्कोपी फोटो इज शाउटिंग एट एन इमेज इन फ्रंट ऑफ यू, then you can't ignore. I mean, that is the mainstay of diagnosis. When something has been confirmed on microscopy, then minor changes in the history you will have to ignore. Okay, ठीक है, done, fine. Someone called me an alien. Good or bad? I hope good. Okay, anyway, thank you if that was good. Now coming back, there is a 26-year-old male from a rural village with history of swimming in the pond, presents to the physician with complaints of fever, headache, confusion and vomiting. Okay, not getting distracted. So there is a patient who's come to us with a history of Yeah, rural मतलब मैम he is taking bath in dirty water, swimming in pond, बिल्कुल नाले वाला पानी. And now he's having all meningitis kind of complaints, parasitic infection लगा, culture करना है. अब ये what did you think of नाला नहाना मैम? नाइग्लेरिया तक तो मैंने सोच लिया, but they asked me the culture media. So now filling in the final part of the table, whether they write naigleria or they write acanthamoeba, it is non-nutrient agar. What is NNA? It is going to be non-nutrient agar. For both of them, it is the same culture media along with thoda sa, because non-nutrient, you have not added any nutrient, so you supplement it with what? You supplement it with E. coli. So what all have they written over here? Non-nutrient agar with E. coli is what is going to be used over here. Okay, for both of them. यहाँ पे तो naigleria है, but even if it would have been acanthamoeba, answer would have been the same. Perfect. Moving on to the next one. Sabka favorite. 25-year-old patient presents with fever. Again, back to everyone having diarrhea, nausea for 3 days. History of foul smelling fatty stools. Patient is having steatorrhea. Steatorrhea means patient is having malabsorption features. When malabsorption comes, you have to think of Giardia lamblia. Otherwise, the patient is vitally stable. Stool examination shows a motile protozoa. Ma'am, Giardia, girta hua leaf. What kind of motility? Falling leaf motility is going to be seen. Which of the following statement is incorrect for the suspected organism? What did you write? C and D. You are saying C is also incorrect. String test? नहीं, ये तो ठीक है. The incorrect statement, it doesn't have 4 flagella. It has 4 pairs of flagella, which means it has total 8 flagella. So अब what do you call Giardia? What do you नहीं? What do we in a rapid revision call Giardia? He's the Dadaji. He's the grand old man of the intestine. Dadaji of the intestine. So we always used to study that Dadaji is going to be found in the duodenum. Dadaji is going to be found in the duodenum. And duodenum, matlab Dadaji, old people of your family love to go for walks. They're not like us that sit for 3 hours on Netflix and watch movies, and that's our definition of chilling. Their definition of chilling is subah sham walk pe jana hai, right? So they will always be roaming around in the lumen. They will ask you that Giardia is an intracellular organism or a luminal organism. Giardia will always be roaming around, na, like Dadaji. So that is going to be luminal organism. Obviously, I'm sorry I'm comparing, but Dadaji is causing what? Dadaji is causing diarrhea and fatty diarrhea. That is going to be malabsorption. So now what is the infective stage? Ma'am, you told us for everything the infective stage is going to be the cyst. Now, where is the cyst going to be seen? Obviously in the diarrhea sample, in the stool sample. Now let's look at the trophozoite. When I look at the trophozoite, I call him Dadaji. Dadaji has 2 eyes. So trophozoite has how many nuclei? Trophozoite is going to have 2 nuclei. And how many? Some people say Dadaji looks Chinese after this. How many pairs of flagella? Pairs of flagella is going to be 4 pairs of flagella, which means total of 8 flagella. This was the trophozoite. What about the cyst? How many nuclei are present in the cyst? There are 4 nuclei that are going to be present in the cyst. Where did you see the trophozoite and the cyst? Ma'am, in the diarrhea sample. But examiner ne bola diarrhea khatam ho chuka hai. Patient is saying now I'm not having stool sample since the past 2 days. I have not undergone any defecation. So now where will you collect the sample from? So I knew Dadaji was in the duodenum. So I will catch Dadaji from the duodenum. For catching the Dadaji, you will find a dhaga. Dhaga wala test is for catching Dadaji. What dhaga? You are going to send a string inside. Where will you send the string? Ma'am, I will send the string into duodenum because that is where Dadaji is sitting. And now in the duodenum, via this string, I will catch Dadaji. So now coming back, if I say that cyst is the infective stage, that is correct. String test is done from duodenal contents, that is correct. And what is the drug of choice? It is metronidazole, tinidazole. They say clinically tinidazole 2 gram is more effective, but metronidazole, tinidazole. So is metronidazole the drug of choice for treatment? Yes. Now coming back, because you said that there is a motility, which motility are we dealing with? This we know, Dadaji, Giardia lamblia, girta hua leaf, falling leaf motility. How many flagella? Once again, 4 pairs. Don't mess up. 8 flagella. String test abhi to aaj start hua hai. Kal to I will tell you 2 more string tests and then I will make a table. Today I have told you a string test for Giardia lamblia. Now, this was one organism with flagella, very commonly, because now listen carefully, Dadaji ka shape, although you are calling him again and again like a human being, Chinese Dadaji, what all we didn't call him, right? But then they say we want to change it. We want to call this trophozoid as tennis racket appearance. Unka nahi man hua ye Dadaji story batane ka. They called it tennis racket. Now in patho, did I teach you? There is one disease in which you see the tennis racket Burbeck granules. Burbeck granules is going to be, ma'am, wo Langerhans cell histiocytosis was also Burbick granules showing you tennis racket, which you taught us in patho. Now tennis racket appearance is somewhere seen in Dadaji also, that is Giardia lamblia. Very commonly you confuse this with this organism. What is this? In our rapid revision videos, we have called her Dadaji's girlfriend, because she belongs to the same family. Dadaji also used to look somewhat like a tennis racket, and Trichomonas vaginalis also looks like a tennis racket. Dadaji also had flagella coming out of it. Trichomonas also has flagella coming out of it, but they are very, very different. Dadaji causing diarrhea, whereas Trichomonas vaginalis automatically tells me it's a sexually transmitted disease. Remember TTT. Trichomonas only has trophozoite stage. So whether it is what you're seeing under the microscope, it is trophozoite. What is infective? Trophozoite. There is no cyst stage that is existing. So please note, what is the trophozoite showing you? Twitching motility. Trophozoite for Trichomonas is showing you twitching motility. Everyone has started calling Dadaji's girlfriend as very, very fashionable. Yeah, that is correct. Very, very, very fashionable, which means that first, fashionable girl means that number one, look at her hairstyle. Ma'am, she has 4 hair in front, and then she has very one long ponytail at the back. So she has 4 hair in front, which means 4 flagella that are anteriorly, and 1 flagella going all the way posteriorly. So how many flagella does she have? She has a total of 5. Dadaji had 4 pairs. She has 4 plus 1. Don't mess up. Thereafter, how many nuclei? Only 1 nuclei. Dadaji was like Dadaji again and again, 2 eyes. So you will remember Giardia had 2 nuclei in the trophozoite. Trichomonas has only 1 nucleus in the trophozoite. What is the motility? Dadaji, Giardia, falling leaf motility, Trichomonas, twitching motility. What is more fashionable about her? What is the kind of jewelry she will like? She will love very, very rich diamonds, and she will love doing a lot of makeup, putting eyelashes. So diamond media, lash media, that is very, very important. Diamond media hoga, lash media hoga. Which kind of fruit is she going to eat? Very basic fruits like any of us? No, ma'am. She will eat exotic fruits like strawberry. This though you don't even need a mnemonic. It shows you strawberry cervix, which you can see over here, and strawberry has that green color leaf on top. So the female comes to you with a green color discharge, and strawberry cervix is Trichomonas. Treatment, Dadaji ka was also metronidazole for Giardia lamblia, diarrhea ke liye yahan bhi metronidazole, but this is a sexually transmitted disease. So over here, partner treatment is very, very important. Now what I'll want you to do is make me a table and fill in the blanks. ठीक है, टेल मी गाइस, सबका मुझे न्यूक्लियस का नंबर बता दो. आई हैव डन माय जॉब, नाउ यू डू योर जॉब प्रॉपर्ली. टेल मी, एंटमीबा हिस्टोलिटिका का ट्रोफोजॉइट हैड 1 न्यूक्लियस एंड सिस्ट हैड 1 to 4 nuclei. Okay, Entamoeba coli ka trophozoite also had 1 nuclei and cyst had 1 to 8. Belantidium coli is the only organism where trophozoite and cyst both have 2:2. I'll write it as 2 equal to, both of them have 2. Giardia lamblia, like the Dadaji. So the trophozoite has 2 eyes and the cyst is going to have double, that is 4. Trichomonas, the trophozoite has 1 nucleus and cyst with a not applicable because cyst form does not exist. So we are done and the nuclei are settled. Finally, this is your list. We can move forward. Yes, we are still, oh, we have 12 more minutes left. I can do so much more in this. Let's go forward. 45-year-old patient from South America presents with fever, fatigue. Further history reveals being bitten by an insect in the past. On examination and investigation, features of cardiomegaly, which is the organism and the vector that is about the disease mentioned. Some of you said Trypanosoma cruzi and sandfly. Do you want to reconsider? Because you're right with Trypanosoma cruzi, but that is not caused by sandfly, that is caused by kissing bug. Sometimes you know the answer, but in that jaldi wala thing, you end up messing it up, and that is exactly what you don't have to do in the exam. See, there are 3 very classic rules of the exam. Rule number 1 is that your gut feeling is always the correct answer. When is it not? 2 exceptions to this rule is, सबसे पहले तो gut feeling से जो first answer लगता है उसको answer mark करना. But if that answer is option A, give yourself 5 extra seconds and read through all the other options to be extra sure. If that answer is to do with pedigree analysis and you have answered it within 5 seconds, then also stop. Because probably your gut feeling in pedigree charts has gone wrong. So two places where your gut feeling can go wrong is either it's option A or it's for pedigree chart. वहाँ पर थोड़ा overthinking करना. Otherwise, overthinking is not required. Your gut feeling is usually the correct answer, which I'm telling you. ठीक है, I'm telling you very crude methods. When you've given up hope in the exam, and there comes a point in the exam जब समझ ही नहीं आएगा whether you studied MBBS or you studied BTech, MTech, क्या पढ़ के आए थे लाइफ में, because the question will look so alien to you. You will not even understand whether it is from your course syllabus or not. And that is when these kind of hacks will help you. Now coming back, how can you get Trypanosoma cruzi? Now anyone getting this wrong is like, I take offense. इतनी अच्छी story बताने के बाद you do this wrong and I will only not forgive you. You cannot do this wrong. I mean, you owe me this one in the exam. If it comes, it's not for you, it's for me that you have to get it right. When I'm talking about Trypanosoma cruzi, I'm talking about a very, very rich couple who's from America. So this is known as South American sleeping sickness. Many of you have focused only on African sleeping sickness, which I will talk about later. This is South American sleeping sickness that you have to go on. And South American couple will be very rich. The rich couple will have a Mercedes Benz G-Wagon, will travel on a cruise, will go to Rome, will lead the most jhakas life always. And of course, we will taunt such a friend of ours saying, Bade log badi baatein. If any friend of ours end up living such a life, then of course he must be not be a doctor. And then he will end up being called as this. But before calling Bade log badi baatein, we have the taunting thing. Taunting thing is, bade log badi baatein. Now, why did I tell you this story? Because the cruise will remind you of Trypanosoma cruzi. The jhakas life will remind you of Chagas disease that this patient is suffering from. The bade log badi baatein will tell you that is the NNN culture media on which we grow Trypanosoma cruzi. Bade log badi baatein because all the organs become big. There is mega esophagus, mega colon. Even if they said cardiac, there is going to be cardiomegaly. All organs become big. Then Rome, that's the most favorite destination of a rich person. Rome, Italy, so and so. So Romana's sign. What is Romana's sign? It is unilateral, usually swelling. Or in other words, if they don't want to write Romana's sign, it is periorbital edema is how they are going to cause it. And how will you treat it? Benz. What is Benz? The drug of choice is benzimidazole. Benzimidazole. Yes, that is correct. Now come back. If you had to mark, you had a patient from South America. That is number one. Next, bade log badi baatein. So cardiomegaly. Now you know it's cruise, and cruise is going to be caused by what? So what do you think? This South American couple is living such a lavish life. What have they gone for, ma'am? They've gone for their honeymoon. And whenever they think of honeymoon, you will obviously think of kissing. But here I've converted it into an insect known as kissing bug. The other name of kissing bug is what? Triatamine reduvid bug. Here I ruined it, the entire story, but you'll have to learn it. So triatamine reduvid bug is referred to as the kissing bug, and that is what they've spent so much of money for. That is their honeymoon. So cruise ke sath you will think of kissing bug. Many of you marked sandfly. No guys, sandfly jo karata hai, that is kala azar, that is leishmania, totally different. What else is written over here? I have one more thing written. Trypanosoma brucei and CC fly. Trypanosoma brucei and CC fly is what is going to cause African sleeping sickness. And there are two types of African sleeping sickness. One is known as West African sleeping sickness and the other is East African sleeping sickness. The only question that comes from sign point of view, which of them, तो ये तो पता लग गया मैम, African वाला sleeping sickness is caused by Trypanosoma cruzi and that is the CC fly and that is West and East. Someone asked me winter bottom sign. You are telling me, tell me the pura question na. Winter bottom sign that you're telling me is what and where is it seen? West African or East African? Winter bottom sign, W for W, is seen in West African and it is lymphadenopathy. So if they say there is a female or a male with a big lymph node which is swollen over here on the neck, they are telling you winter bottom sign in West African sleeping sickness. On the other hand, they talk about this swollen eye, Romana's sign. The Rome is a rich couple, jhakas life, so that is Chagas disease, Trypanosoma cruzi. दो ही signs हैं. Romana's sign is cruzi and Chagas disease and winter bottom sign is West African sleeping sickness with a lymph node. Moving on to the next question that we have over here. There is a man with fever of unknown origin or pyrexia of unknown origin, has a job that requires traveling around the world. The bone marrow picture has been shown below. Which of the following investigations would be the most appropriate in this case? And A, everyone is saying. What are you going in order, by the way? Ke Cruzy kara, Brucey kara, to Leishmania karenge. But anyways, you're right. RK39, that is Leishmaniasis. Now let me tell you one very important thing that will trouble you in the exam. Whenever you get a macrophage, and I'm going to draw a big nucleus, ye hai aapka macrophage. Inside the macrophage, whenever you will see organisms, if they ask you about a fungus, which organism is going to be present inside the macrophage? Reticuloendotheliosis, histoplasma. The second one, if they talk about a parasite, it is going to be Leishmania, matlab Kala Azar. Under the macrophage, these are the two favorite histories that they want to give you. And ma'am, this what you see, I want to show you another picture which I can zoom into it. We all know how does Kala Azar present. The reason, quite racist, but they called it Kala Azar only because the child's skin or the patient's skin becomes hyperpigmented. So this must have been a name kept in the olden days. They named the disease only after it. That is referred to as Kala Azar because of the hyper skin pigmentation that happened. And apart from that, you will see that there is a protuberant abdomen because of organ involvement, hepatosplenomegaly. Yesterday or day before only I was teaching you that whenever there is a massive spleen and in the MCQ exam you are dealing with such a situation, either you will think of Kala Azar. Apart from that, which leukemia? Philadelphia wala CML. Apart from that, remember the TikTok boy, hairy cell leukemia. Hairy cell leukemia involves red pulp of the spleen or white pulp of the spleen. Sab kuch pada hai. I am just confused. I'm just adding on to your knowledge. That is what, ma'am, hairy cell leukemia, red hair TikTok boy, red hair was or red pulp was getting affected. Very good. So this is massive spleen. Malaria also. This I'm not saying massive spleen has 1 million things. I am telling you exam ka buzzword kya banega. So massive spleen is seen. Kala Azar means Kala patient. So now we come to what do you see under the macrophage. When I zoom in, I can see big dot, small dot, big dot, small dot. Histoplasma just showed me 1 dot. When I start seeing big dot, small dot, the big dot is the nucleus, the small dot is the kinetoplast. This is kinetoplast. So which means together they are known as LD body. Obviously Leishmania donovani will show me LD bodies. What is the treatment that you do for Leishmania donovani? What is the treatment for Kala Azar? I mean, what a stupid question I'm asking you. It is liposomal amphotericin B. Yes, everyone says that. But if I ask you what is the treatment of post-Kala Azar dermal leishmaniasis, after Kala Azar, if there is, you can see these hypopigmented lesions, dermal leishmaniasis, then it is known as oral miltefossin. ठीक है, this all you know from pharma, you are good at anyway. Coming back, if you had to do a rapid test, ma'am, क्या test करें? Test जो करते हैं, that is RK39. See, every test, every disease has a card test. Malaria का card test का नाम मलेरिया, कोविड के card test का कोविड, HIV का HIV, UPT का यूरिन प्रेगनेंसी टेस्ट. काला आजार के कार्ड टेस्ट का नाम याद कर लो. इट इज नॉट काला आजार कार्ड टेस्ट, इट इज RK39 कार्ड टेस्ट, और यू कैन से RK39 इज द एंटीजन. आई हैव लर्नड इट एज इट इज कार्ड टेस्ट है ना, तो इट इज अ रैपिड टेस्ट फॉर व्हाट? इट इज अ रैपिड टेस्ट फॉर काला आजार. सो RK39 is the antigen testing that we have for Kala Azar, which was the answer over here. Now I move on to the next question, and everyone's favorite also. You have a 36-year-old known case of HIV. CD4 count is like almost next to death only, very low. He presents with complaints of diarrhea. HIV positive patient with diarrhea, I will always think of coccidian parasites. I will always think of coccidian parasite. Yes. Acha, before that, someone is asking me, ma'am, where should we take out the, sorry, I'll have to go back to the question for that doubt. Where will we take the sample from? Okay, that's quite a controversial question. But if you ask me which is like the most sensitive site, कहाँ से sample आना चाहिए, answer is spleen. But practically सोचो ना, do you want to puncture someone's spleen for the diagnosis of Kala Azar? Because spleen is a very vascular organ. I put a needle into it. What if I can't control the bleeding from the spleen and patient dies? I mean, I'm pretty scared to do that kind of a procedure. Then after that, I go in for bone marrow. I mean, bone marrow is also very painful, but at least bone marrow will not have that kind of a complication of bleeding to death. So bone marrow is also good, but bone marrow में भी नहीं मानता patient, कराना ही नहीं है उसको, then blood sample देगा, और क्या option बचा? But again, that's the order which we follow. Then I come back to the question. Yes, what did you mark all of it? All of you? I was getting a lot of DD wala options, and those who didn't want to answer D said Neeta Ambani. समझ जाओ ma'am. it is our internal thing. So when I think of HIV positive patient with diarrhea, I said coccidian parasites. Coccidian parasites, we write it as 3. Number 1 is going to be Cryptosporidium. Second one is Cyclospora and Isospora, and I've written it in an alphabetical order, and you know you have to write it this way, otherwise you can't learn it. So Cryptosporidium हो गया, फिर CR, फिर CY Cyclospora. Isospora का नाम चेंज हो गया, माइक्रोबायोलॉजी में फेवरेट है, जब कुछ नहीं समझ आ रहा नाम बदल दो. सिस्टो आइसोस्पोरा, इट इज द सेम थिंग यार, आइसोस्पोरा लिखे, सिस्टो आइसोस्पोरा लिखे, चलो सिस्टो आइसोस्पोरा भी लिख लो, देन ऑल्सो दिस इज अल्फाबेटिकल ऑर्डर ओनली. After writing, now you will tell me what is the stain that has been used over here, because you told me that for coccidian parasites, ma'am, you had to do not just the Zn stain, you had to do the cold Zn stain, which is also known as the Kinyon stain, and that is what you've done over here. Okay, now when I look at the size, Cryptosporidium is 4 to 6 microns, फिर double करना और triple करना है. 4 का double is going to be 8, 6 का double is 12, so Cyclospora 8 to 12. Then you take the 12, double is 24, and the triple is 35, 36, so 35, 36 microns tak. So first you double it, and then you are going to double and triple it, that is Isospora. Talking about the shape, Cryptosporidium you write with the C, so just complete it, the shape is going to be round. Cyclospora you write with the C, so the shape is going to be round. Isospora you write like an I, so the shape is going to be oval. Round, round, oval. Now which of them are acid fast? All of them are acid fast, ma'am, but Cyclospora is a cycle. Kabhi haan, kabhi na, cycle. Sometimes acid fast, sometimes not acid fast, which means it is going to be variable acid fast. Is that okay? So let me see the photo. Number 1, the best way of identifying the photo is ये round है, ये round है, तो ये तो CC वाले organisms हैं. This one is coming out to be oval, so if it's oval then it is Isospora, तो Isospora तो सबसे easy हो गया. Coming back, CC वाले organisms, कौन सा Cryptosporidium, कौन सा Cyclospora? So Cryptosporidium तो everything is coming pink. Fully acid fast, this is Cryptosporidium. On the other hand, Cyclospora, some of them are coming acid fast. If I zoom in further, some of them are not, they are white. This is also not. So sometimes yes, sometimes no. Cyclical pattern show कर रहा है. Cyclical pattern वाला Cyclospora. So I've made a cycle next to it. You must know Cyclospora is cyclical. Cycle पे क्या खरीदने गए? I mean, our mothers used to send us to get a packet of milk. Now, of course, your mothers don't send you because you have Blinkit. But I have made it all the more. Next level exotic. Cycle पे क्या लेने गए? Raspberry लेने गए. So what is raspberry लेने गए? Because if they always tell you that there is an HIV positive patient with diarrhea, but the diarrhea has happened after consumption of raspberries, that kind of a history is going to be associated with Cyclospora. ठीक है? Cyclospora का raspberry. Cycle पे Cyclospora. Cycle पे we have gone to buy raspberry. Now coming back, treatment for Spora brothers, Cyclospora, Isospora, Spora brothers के लिए it is going to be the cotrimoxazole. Whereas for cryptocurrency, that is what is Neeta Ambani that everyone was saying. Neeta's oxanide is the drug of choice, and that is exactly what we had written पहले. The infection is self-resolving. पहले तो कौन सा बनाया? 4 to 6 microns लिखा था. Crypto, Cyclo, Iso. सबसे छोटा वाला Cryptosporidium. So what is the treatment? Such as Neeta's oxanide is given. I think then I don't need to read the other options. It is pretty obvious. Now coming to the INI-CET question. A crypto मतलब hidden. Hidden, don't, everyone has to listen, this can come in NEET and FMG also. Crypto matlab hidden, so ma'am, when it goes into my intestine, it does not nicely go and sit inside the cells. It says I will hide, like I'll make a tunnel, एक tunnel में छुप के बैठ जाऊंगा. Cryptocurrency ना, hidden रखना पड़ेगा. So now when I look at the intestine, I can see that this parasite is hiding like this. See, it could have gone inside the cells and chilled, relaxed. It said no, I will go and sit. If you look over here, why is this intestine? ये कौन से वाले cells हैं guys? These are the goblet cells, and I can see that oh, this is hiding in the brush border. So Cryptosporidium tends to be present along the brush border. वो tunnel जो उसने बनाया for hiding, what is the name of that tunnel? Parasitiforous vacuole. So parasite का vacuole, parasitiforous vacuole. Who is hiding? The cryptocurrency is hiding. Now exam में I'll tell you the error that you do. Right now you will say इतना stupid नहीं करेंगे मैम हम, but in the exam you will do it when you are stressed. Cryptosporidium and Cryptococcus को same पढ़ के आते हो. Cryptosporidium is a parasite which is causing diarrhea in an HIV positive person. Cryptococcus was a fungus which was causing meningitis in an HIV positive person. It's very obvious. But in the exam, you might write the treatment of cryptococcus as Nita Ambani. And no guys, nitazoxanide not for cryptococcal meningitis. I'm talking about cryptosporidium. Please exam में आंखें खोल के देखना. Crypto क्या? Cryptosporidium is what I'm talking about over here. Don't do just crypto Nita like that in the exam. So now coming all the way back, let's give you a quick question again. How to identify this organism which has a 25 micron Kinyon positive oocyst in an HIV positive patient with diarrhea? You can see this oval Kinyon positive मतलब cold Z and stain, largest size 25 micron. क्या mark करोगे? Isospora. But ma'am, Isospora नाम नहीं लिखा. Isospora का क्या नाम लिखा? नया वाला नाम, same thing. Cysto isospora, iso cysto sub same. ठीक है, done with this also. Coming to one more question. I think we're left with just two more for the day. Identify the life cycle that has been shown over here. Human being तो हर किसी के life cycle में होता है, उससे तो we can't identify. We will have to look at other animals. I see a cat. As soon as we see a cat, what comes to my mind is Toxoplasma gondii, because Toxoplasma में we always used to learn it that by making a cat. Definitive host in Toxoplasma is going to be the cat, and as soon as I see the cat, my answer is ready. So what happens in Toxoplasma? Toxoplasma gondii में there are three states, and then you know that brain का calcification वाला question that ma'am will ask in the end. सबसे पहले Toxoplasma gondii के तीन चीजें होती है. Which of them is infective? You will answer. Bradyzoids are infective to us, tachyzoids are infective to us, or oocyst is infected to us. बताओ जल्दी. Tachyzoid, bradyzoid और oocyst. Take one answer. All. Okay, you say ma'am it is all of the above. Don't trick us. Okay, perfect. It is all of the above. What is infective? All are infective, but root is different. So when we say cat, do you know what I'm going to teach you? Till now I was teaching you our diarrhea, human shit. Now I'll teach you cat shit. I will write it as shit only so that you remember. तो cat का जो feces होता है, cat उसके अंदर क्या पास करती है? cat पास करती है oocyst. Is oocyst capable of causing infection? If oocyst mixes in our food and water, can it come and cause infection? No. This means that that oocyst has to have sporules inside it. Once the organism will form, then that can come and cause infection to me. So ma'am, what is going to happen is it will take time. You need to have sporulated oocyst. So now my question to you is new and fresh cat shit infectious to us, or is old cat shit infectious to us? So it is the old shit that we are scared of, because the new shit only has oocyst and that is not infective to us. It is the old shit which is infective to us, and that is going to come as fecal-oral route, and that is why in the bracket you will write old shit. Okay, what are you scared of? I'm scared of old shit coming in my food and water. Very important. That is one way of getting the infection. Second way is either we can get it via blood transfusion. Someone was having the disease, his blood came into my body, so blood transfusion. Second is tissue. Maybe imagine eating the tissue of some animal and that animal has this disease. So one is bradyzoid, one is tachyzoid. BT is what you have to go alternative. For example, if they ask you how do you get tachyzoids, so we get tachyzoids via the blood. How do we get bradyzoids? We get bradyzoid via the tissue. So if they say animal eating से हुआ है, tissue से हुआ है, you will say bradyzoid. If they say अच्छा blood transfusion से हुआ है, you will say tachyzoid. Let me twist the question. If they say this has happened following organ transplantation, organ transplant from the organ is a tissue, which form would have come? It would have been bradyzoid. If they say it has happened mother to child, ma'am, mother to child तो blood goes na, so mother to child, if blood goes, what will you mark? You will mark tachyzoid. So they will not write blood tissue because you have read BT. They will give you a situation. Just fit it into either blood or fit it into tissue and go BT. Is that okay with everyone? Toxoplasma is done. Now comes the question that you know. Toxoplasma, let's assume mother to child chala gaya via the blood, the tachyzoids have gone. Now the child will have what kind of calcification in the brain? Child ka calcification in the brain. Is it periventricular calcification, cerebral calcification, parenchymal wala, या फिर gray matter, white matter? जल्दी quick. Someone meanwhile asks me definitive host, intermediate host. कल बना ले Dr. Lakshita. Maybe I can give you as the bonus slides. That would help you, like, because then there's nothing for me to tell you in that. It is a reading out things I'm not doing in LRR. Those I'll add in your bonus slides. Okay, perfect. When you're talking about Toxoplasma, ma'am, it is parenchymal, just cerebral calcification that you will say. Tell me the others also. ऐसा एक single मत लिखो. What will you see? CMV में क्या दिखेगा? CMV में you will say periventricular calcification. Very good. And tell me one more thing. When you see, I'm writing short form, gray matter, white matter junction. Gray matter, white matter junction पे if you see calcification, which virus will you think of? Zika virus. Did I get that answer? Yes, I got it. Perfect. ये ब्रेन में रेडियोलॉजिकल कैल्सिफिकेशन, remember this, most important chances, cytomegalovirus and periventricular calcification, toxoplasma is pura cerebral calcification, gray matter white matter junction calcification, Zika virus. Okay, done with this. I think I'm left with exactly one question more for the day, and then you can go and have your lunch in case you have not. 32-year-old patient again from rural India with fever and chills. Fever and chills सोचते ही I think of malaria. And in the last class I taught you कि malaria का blood count करोगे तो what count will be more? Neutro, lympho, mono, EO, baso. Monocyte will be more. तो देखो monocyte is found to be elevated. मैंने fever chills के साथ malaria सोचा plus monocyte elevated. Peripheral smear with JSB stain. As per the malaria program of PSM, what is the stain that we should be using for malaria? It is the same hemat wala stain, but only named after the scientist, that is Jaswant Singh Bhattacharya stain. So ma'am, actually ये stain होता क्या है? Is this the same hemat wala stain? Hemat wala कौन सा stain? Romanovsky family of stain. Yes, it's the same. The only thing for INI, पहले तो सबके लिए याद कर लो, Romanovsky stain होता है, उसका only scientist version they have put up for malaria. It's the same Romanovsky stain. Now they call it Jaswant Singh Bhattacharya stain. No issue. But for INI-CET exam, they will say Romanovsky stain you use in hematology, that has a pH of what? That has a pH of 6.8. The same Romanovsky stain, if I want to visualize malaria, I want to increase the pH a little to 7.2. फिर stain का color becomes better if you alter the pH. Of course, this information is only for INI-CET students. अब पूरी स्टोरी खत्म. नाउ आई कम टू पेरिफेरल स्मेयर शोइंग मी बैंड शेप. मैम बैंड, माला, माला बैंड, प्लाज्मोडियम, malaria. So what are you going to do? You are now going to see all the photos before you end today's class. I have given you the photos of all the ring forms. ये vivax का ring form, falciparum का ring form and malaria का ring form. What is ring form known as? Ring form is known as trophozoite. So live simple. When I write vivax, so you will say ma'am one ring, one dot. Everyone, vivax, one song karo, song of the day. Vivax, one ring, one dot. So if I zoom in, I will see one ring and one dot inside it, exactly over here. I'm not overdrawing, I'm drawing on the side. So that is vivax. But when I look at falciparum, opposite, ma'am, look at this, many rings. So many rings I can see, and I can see two dots, many dots. So many rings, many dots. That is known as falciparum. Some people now run their imagination. They say, अच्छा ये ऐसा two dots बन रहा है. This is looking like headphones. This is looking like speaker phones. So your wish, whatever you want to call it. But then so many rings came that one of the ring decided to become the Fufaji of your family, got angry and said, oh, you are not giving me space. I'm going and sitting in one corner. Who is that ultra dramatic ring? That ring is known as the Acol form. Acol or applique form is that angry Fufaji of the family who says that, oh, arrangements are not good. I am going and sitting with a big face in one corner. So can I say the ring that has gone at the margin and has gotten angry, that is what? That is the Acol or the applique form. So this is the Fufaji form. So where do you see Fufaji form? That Fufaji, face swollen, angry always form is seen in Plasmodium falciparum. Finally, coming to the rings of malaria. Ma'am, malaria ka ring is not looking like a ring. If this is a ring, it has become a pichkawa ring. It has become a flat ring, or it has basically become a band. So now I say mala band, Plasmodium malaria, mala band is how you are going to learn it. Simple. If they say one ring, one dot, vivax, many rings, many dots with Fufaji, angry, Acol, applique form, falciparum. Mala ke liye band form. All the bands and all the rings are done. Now we come to gametocytes. Vivax ka to gametocyte is going to be oval. I can see that oval. Whereas falciparum ka gametocyte is banana shaped. Now instead of banana shaped, they want to write crescent shaped. They want to write elongated shape. तो यही बनेगा, सब कुछ सेम. Now the question is, when I use the word macrogametocyte, it means female, and when I use microgametocyte, it means male. Not in every plasmodium. In every malaria, macro मतलब females को ज़्यादा importance मिलेगा ना. I mean obvious. Macro is going to be females and micro is going to be males. यहाँ तक समझ आया. But now in Plasmodium falciparum, they ask you which out of these is this cluster. Is this male or female? Male or female? मैम banana shaped है, falciparum है, समझ आया. This is, and then there's another cluster over here. So please note, you will have to look at that brown color ka nucleus. If you look at this nucleus, ma'am, if all the nucleus is very well organized, like girls keep their stuff na very nicely, you will look at their study table, their books will be in one corner, pencil stand alag hoga. Then they will have different sections. Look at their copies, they have different कलर कोडिंग पेजेस, उनका अपना कलर कोडिंग वैक्यूटेनर जैसा चलता है कॉपी में, पिंक कलर हाइलाइटर, येलो कलर हाइलाइटर. सो गर्ल्स कीप एवरीथिंग वेरी वेल ऑर्गेनाइज्ड. So if the nucleus is very well organized, this has to be a female gametocyte. And then come boys. With one blue color pen, they have made all 19 subject notes. Ma'am, everything is scattered. Wahi books hai, books ke upar I put a sheet, it becomes a pillow at night. So that's how the boys hostel room is. Everything is scattered everywhere. Okay, sorry for being very relatable, but no other choice. So everything is scattered everywhere, and that is known as a male gametocyte. Now you will never ever get it wrong. Okay, so male and female is settled. What is left? Coming back, यहाँ पर तो माला था, माला band was very very classical. Now you have been answering some random things, which is the next question. 32-year-old patient from rural India again, same fever and chills, मतलब malaria, monocyte increased malaria. Peripheral smear shows crescent shaped gametocytes, banana shaped, crescent shaped, falciparum. What other finding is seen over here? Yes, what other findings are seen over here? Okay, next A, B answers. अच्छा, falciparum में क्या दिखता है? Morer's cleft. So I think this is what you have to study. Very simple. No one will ask you the photos for this, but names will be asked. तो अब एग्जाम में क्या होता है? वाइवा में तो मैम जैसे आप हमें पढ़ा रहे हो ना टॉपिक बाय टॉपिक, ऐसा तो एग्जामिनर पूछता ही नहीं. वाइवा में इट्स शफलिंग. फ्रॉम बैक्टीरिया ही विल आस्क मी मेडिसिन, फ्रॉम मेडिसिन ही विल आस्क मी रेडियो. तो वाइवा में सब कुछ शफलिंग, तो वाइवैक्स में शफनर्स डॉट्स. Plasmodium falciparum, ulta pado, MAUR, it will show you MAUR, Morer's clefts. Falciparum will show you Morer's clefts. Next, Plasmodium malaria will show you man dots, Z man dots. Malaria, man dots. And ovale जो बचा 5 अल्फाबेट वाला, उसका जेम्स डॉट 5 अल्फाबेट वाला नहीं भी याद करोगे तो वॉट एवर इज लेफ्ट. तो वाइवा में शफलिंग, falciparum, Morer cleft, malaria, man dots, and ovale is James dot. So all these are settled for us. Truly and genuinely last question for the day, because I keep tricking you on that, but ये सच में last है. Which statement about Plasmodium species is incorrect? अच्छा, someone said OA James. ठीक है, very nice, OA James. ऐसे याद कर लेते हैं. Coming to which statement about Plasmodium species is incorrect? Jaldi, last question of one question, one photo. Can you do it in 1 minute? One question, one photo. Okay, everyone is saying option B is incorrect. Okay, let's see. Drug resistance falciparum is seen in India. Ma'am, India is not for beginners. Drug resistance, everything is seen in India. तो ये तो मान लेते हैं. Okay, severe malaria क्या होता है? That is severe malaria with shock is called algid malaria. Yes, I want to tell you, when I say malaria, you know, algid means that the patient has gone into circulatory collapse. Circulatory collapse or circulatory failure. In simple words, it's a malaria patient that has gone into shock. तो मतलब ये भी correct है. Relapse is seen with Plasmodium ovale. What is relapse and how is it different from recrudescence? Relapse और recrudescence में क्या फर्क होता है? Relapse का मतलब होता है that let's assume someone had malaria infection, but then that malaria decided that I am a very lazy malaria, तो मैं liver में जाके सो जाता हूँ. So where are the hypnozoites form? Hypnozoites form मतलब hypnotized वाला malaria. Malaria entered into my body and decided that, oh, let me go and sleep off in the liver. One day I will wake up. That reactivation is known as relapse. Versus what is recrudescence? Recrudescence means that you gave treatment to a patient, but most Indian patients decided after one drug they will not consume. That is inadequate treatment के बाद जो आता है, that is recrudescent malaria. Repeating, relapse is the malaria is sitting inside your liver and then one fine day wakes up again. Recrudescence is that the patient did not take the treatment. So please note that relapse is going to be seen with vivax and ovale, and recrudescence is seen with falciparum and malariae. So how do I learn this? Because this is a very epidemiology PSM kind of thing. छोटा वाला नाम show you छोटा वाला नाम. Vivax and ovale show you relapse, whereas falciparum and malariae are going to show you recrudescence. So tell me, does ovale show you relapse? Chota naam, chota naam? Yes, it does. The only wrong statement is this: presence of Duffy antigen is protective against Vivax. No, please note, when Vivax enters into our blood, Vivax always enters via a door on our red blood cell. That door on our red blood cell is known as Duffy antigen. That door is known as Duffy antigen. It only has to come in via Duffy antigen. So Duffy antigen, okay, Duffy makes you dumb. It is Duffy which has actually created a problem for us, but we all have Duffy, so we will have Vivax susceptibility. There are some people who are born without Duffy. They might feel that, oh, we don't have Duffy, oh, poor us. It is not poor you, it is lucky you, because now you will not even have Plasmodium vivax infection. So the presence of Duffy antigen is not protective. It is actually, in other words, the absence of Duffy antigen which is going to be protective. Someone who does not have Duffy antigen will not have Vivax infection. I think that is settled. Before I end, plasmodium कितनी सारी बात करी, so many ring forms you spoke about. If I अच्छा first answer this much, ये photo होता तो क्या बनाते? No bias, one ring and one dot. What would you have made? Ma'am, I would have made Vivax, one ring and one dot. But जैसे नीचे वाला photo download हुआ exam में, I saw that four rings were coming together. Very good. What is this? This is Maltese cross appearance. Maltese cross is nothing but four rings coming together. That is a tetrad of rings, and that is caused by Babesia microti. Can you tell me which animal is going to be responsible for this? Ma'am, this is reservoir. कहाँ से आता है? Rat से आता है. Can you tell me what is the vector of it? It is microti, micratic. याद रख लेना. From today onwards, you learn it as micratic because rat से आता है, tick से आता है. That is a hard tick. Yes, rat से आता है, tick से आता है. So that is hard tick. Truly, this is the last topic for the day. Now, today at the end of this PDF में bonus में तो you will get all those antifungal, antiparasitic drugs as decided 3 hour 15 minutes usual. We are done. Half of parasito जैसा सोचा था done. Tomorrow first we start. Tomorrow's class is at 12 noon. So we are again same 12 to 3 वाला चलेगा. पहले we will do 30 minutes of helminthology. Then we are going to do bacteriology. Then we are going to do virology. Okay, that is the plan. And yes, what you have for today is you have to make sure that you go back and do the Q bank. So I've made you do most of the PYQs. I've given you most of the revision of images and mnemonics. Now your homework ending is that you have to go and solve the Q bank. So you must do that because that is a self-assessment. Timer के साथ करो. You are now exam going students, which means that you cannot rely on I will do one question, read the explanation for 30 minutes, then do another question. Take up 20 questions, 30 questions, put a timer, finish those, then read all the explanations together. You're giving these mini grand tests in the day without even realizing that you're doing that. So get into the exam mode of solving MCQs. Okay. Yes, some things we have not studied today because abhi nahi khatam hua. Don't worry, we will study tomorrow. What is the association with sickle cell anemia of what? Of what? So basically malaria, see, malaria has to, I think you're asking about malaria only. So malaria has to survive inside a red blood cell. House of malaria is red blood cell. If the house is not stable, how will malaria survive? Imagine a patient has very severe anemia. Hemoglobin RBC is going to break off. Malaria will not be able to survive. Patient has sickle cell anemia, thalassemia. Ma'am, hemoglobin is not there. RBCs are breaking. Hemolytic anemia hai. RBC nahi hoga, home nahi hoga. So how will the malaria survive? So patient might get the infection, but the survival of the malaria is going to be impacted. Okay. Fine. Immunology, Dr. Rohit, I will cover. Don't worry. वो agglutination test, CAT, SAT, MAT, वो साथ साथ बैक्टीरिया के अंदर ही साथ साथ खत्म हो जाएगा. 10 most important bacteria. Don't worry, Dr. Himanshu. What we will study in LRR will automatically be the most. I know you guys are scared of bacteriology. I am also scared of it. Everyone is. So more than you, I am scared कि अरे 1 घंटे में सारे bacteria हो तो 10 hours also is less for bacteria. In 1 hour, how will I finish the bacteria? So the tension is more on me. Leave it to me. For till tomorrow, leave that entire tension on me. I will tell you the most important questions. Galactomannan is a type of a manoprotein only, Dr. Mahima, so not much of a difference. Okay, someone's asking me ectoendopiedra. I don't have photos. Is it okay if I can get that tomorrow? Because piedra, to matlab I will explain it कि वो white dot होता है, but then ectoendothrix are arthrospores. I mean, I can't, without photos, I can't explain that to you. So I will get photos and we'll definitely discuss it tomorrow. Helminth ke sare tricks, don't worry, Dr. Raghav. For helminthology, I've got a photo of all the eggs on one page, all the adult worms on one page, wo Ascaris and Ankylostoma, wo adult worms wala one page, all filaria on one page. That is how we are going to study, right? Okay. Histoplasmosis and sporothrix image, when I see the image alone, but why? There is no similarity. Histoplasma का तो macrophage वाला photo है, whereas sporothrix का तो rosettes वाला photo है. So there should not be any confusion between these two. PDF will be available on the app. You can download it from the app and I will put it on my Telegram group. So whatever suits you. You can get it from the app also, but yeah, within 10 minutes. 10 minutes लगेगा to upload and to send it across. Sporotrichosis वाला हो गया. Sure, I will do that. Color of non-acid fast is it's the same as the background. See, when you're adding methylene blue, that will then go and stain the bacteria which have not been stained with carbonyl fusion. So it's methylene blue wala, blue background. INI topics list, Dr. Vijay Lakshmi, tomorrow, like in patho, I gave you at the end of the 2 days. For micro, also end of tomorrow's you will get. Yeah, so everyone's reminding me to drink water. Thank you, because now I am truly running out of um, this thing, capacity also. Very good, Babesia, azithromycin, atovaquone, you're right on the treatment, perfect. Headphone shape, malaria, no, falciparum. Dr. Prakash, Plasmodium falciparum. Okay, so I think culture media, okay, Dr. Disha, that is bonus slides. वो कल वाले उसमें. After bacteriology is done, your bonus slides will have a list of all hosts. It will have a list of all the culture media also. Done. Anything else that I have missed out or you want? वो अभी कल का भी session बचा है ना, तो वो कर लेंगे. Don't worry about it. And no one's running anyway after that also. There with you till the end. अभी there is considerable exam time है. At least for us to have 1 or 2 more sessions. So we will keep ourselves busy in that entire loop. Don't worry about it. अभी like I'm saying, focus on the most important things. Okay, fine. Thank you so much guys for joining in. Tomorrow I meet you same time. That is at 12 o'clock. Thank you.